progress and change. But as the need for fertility care continues to change, there are challenges we must overcome. As global demographics are changing, fertility rates are under pressure. As technology is advancing faster than ever, clinics and patients still need the tools, time, and training to turn data into informed decisions. And the limited access to quality fertility treatment is a global reproductive issue that needs to change. At Cooper Surgical, we are proud to offer leading IVF and genetic testing solutions to clinics around the world. We believe in pushing the boundaries of what is possible, that together we can change the standards of fertility care to increase the time for patient support and consistently optimize outcomes, change the journey in fertility care to reach a higher level of insight, efficiency, and trust, change the access to fertility care to break down the barriers standing in the way of parenthood. That's how, together with her, him, her, they, them, we can make the change every generation deserves, a chance to bring new generations to life. Cooper Surgical, changing fertility care for generations to come.
Good evening, everyone. I'm Vasudha Bala, working as a product manager for Cooper Surgical India. I welcome you all for today's engaging webinar on clinical value and procedure of sperm function test. This webinar is a part of the educational series direct from Andrology Lab, which has been initiated by IHERA, the International Human Embryology Research Academy, and we at Cooper Surgical are extremely honored to be the knowledge partner for this program. So without any further ado, I would like to start introducing the coordinators for the evening. First up, we have Dr. Rahul Sen. He's currently working as Chief Embryologist in Neelkanth Fertility, Jaipur. He's in the field of ART since last 13 years and is an invited faculty for MIT University for ART Fellowship Program by IFS. He's published various papers in national and international journals. Next up, we have Dr. Sanket Kumal Satya. He's a senior clinical embryologist at Shema IVF Fertility and Reproductive Medicine Center. He's an alumnus of University of Mysore and has done MSc in genetics. He's an ASHRAE certified clinical embryologist and a scientific committee member of IHERA. He's a reviewer of various journals and also authored few publications. To his credit, he's an author and co-author of 3 book chapters. He's a principal investigator for several projects at Shema Medical College. Welcome, Dr. Sanket.
Thank you, Sudha.
Next up, we have Dr. Paresh Makwana. He is an ASRM certified embryologist, and he's a chief embryologist and center head at Wings Group of Hospital, Ahmedabad. And he's also associated with other center of Wings. His key area of interest include embryo biopsy, oocyte and blastocyst vitrification, IVM of oocytes. He's an executive member of ACE India and lifetime member of IFS and IFS Gujarat. He's a trained national and international embryologist without any doubt, and he has served as freelance embryologist in India and West Africa. Welcome, Dr. Paresh. Then we have Dr. Akash Agarwal. He's an MBBS and an ASHRAE certified clinical embryologist. He's serving as scientific director at Hegde Fertility, Hyderabad. He's practicing male fertility and embryology since 2011. He's also a topper at ASHRAE Clinical Embryology, Vienna 2019. He's actively involved in academics and has co-authored a book chapter. He's an active member of various fertility organizations like ASHRAE, ISAR, IFS, and ACE. He's participated in multiple national congresses, and his key area of interest is male fertility treatment with surgically retrieved sperm. Welcome, Dr. Akash. So moving on, I'll hand over the stage to you, Dr. Sanket. You can.
Yeah.
Can you share?
Thank you so much, Vasudha, for that kind introduction. So very good evening to all and warm welcome for today's webinar. So today we have one lecture where Amita Subramanian will be talking about sperm function test and its clinical importance in ART, followed by a series of video lectures where Tejashwini will be talking about sperm vitality, and Madhumanti about sperm DNA fragmentation, and Nidhi Singh about oxidative stress, and Nancy about anti-sperm antibodies, and Lalita with respect to sperm and implant assessments, and Vakil Jagvir Singh about HB assay, and last but not least, Hardik Singh will be talking on automations in semen analysis. So before we start with the first lecture, I would like to invite founder Ved Prakash sir to say a few words.
Thank you, Sanket. So on behalf of iHera and Cooper Surgical, I welcome all of you. We started this recently. We started this program with Cooper Surgical. There are so many lectures, webinar we have done in past, but everyone was all the lectures were based on theoretical part only. So we thought, why not to start the practical aspect of the lab. So in second, we started on sperm function test, and as you know, sperm function test is the baseline. On the basis of sperm function test, you can decide what to do next in the IVF. You can decide what is the status of sperm. And recently, with this, when we started doing ICSI, we slowly forgetting these things, basic things. So today we have Amita Subramaniam. She is going to speak on sperm function test and its relevance in today's practice. So can you please share these slides? And one more thing I want to tell you about that. There is very great news today, and we succeeded to that, the definition of embryologist now changed, and it is it is mentioned in ICMR that all the life sciences are eligible for that embryologist. So now it is applied now, and you all the life sciences postgraduates all are included in that embryology field. So it is very great news, and thanks everyone for supporting us. And my team, ACE team, has done very good, wonderful job on this aspect. So today the first speaker is Mrs. Amita Subramaniam. She possesses 10 years of work experience in the field of ART, currently senior embryologist at Cloud9 Fertility, Chennai since June 2015, and she is IFS certified embryologist. She has various experience in the field. She has done work as embryologist at Srishti Fertility Center, Chennai, and junior embryologist at Sanatani Center of Reproductive Medicine, Bangalore, under the guidance of Dr. D. Swaminathan. And so many presentation and publications she has published in various journal, and she presented as one poster in ASHRAE 2017. All to Dr. Amita, Mrs. Amita Subramaniam. Thank you so much. Thank you.
Amita, please start sharing your screen.
Yeah. Good evening to everyone. Thank you very much, Ved sir, for your kind introduction. So my topic for today's presentation is does sperm function
Sorry to
test?
interrupt, Amita. Can you make it slideshow?
Yes.
My topic for today's presentation is does sperm function tests have any clinical relevance in ART? So today we'll be starting with a brief introduction of what sperm function tests are, and discuss about the need for sperm function tests and the principle of sperm function tests. And then we discuss the types of various sperm function tests available and the clinical relevance of sperm function tests. And then also talk about the emerging technologies available in sperm function tests. So to start with, conventional semen analysis cannot predict the, that is based on the WHO protocol, cannot predict the fertility potential of the male partner with just the concentration, motility, and morphology. And it provides limited information of how well the sperm will perform in vitro and or in vivo settings. And so men with even a normal semen analysis may be fertile or infertile. So sperm morphology per se is not a strong indicator of aneuploidies or DNA fragmentation test. Moreover, the success or rather overuse of ICSI has almost led to the cessation of research on sperm dysfunction and has allowed indiscriminate use of immature or even defective sperm. So sperm function tests, as described by the WHO, is more like a research tool and not part of routine semen analysis in infertility clinics. So this leads us to the question of why do we need a sperm function test? So most of the sperm function tests are mostly performed in academic institutions since clinics are generally not equipped with or rather with the apparatus or the expertise to do these sperm function tests. And Dr. Daniel Franken et al. In 2012 has told us what an ideal sperm function test should be able to permit. So it must allow us the diagnosis of a specific sperm dysfunction. It must allow us to predict the fertilization or pregnancy rates, and it must also indicate an appropriate therapy to alleviate the identified sperm dysfunction. So several studies have also shown that semen parameters of even unprocessed ejaculates and density gradient methods does not affect the ICSI results largely. So it becomes all the more important to identify its sperm dysfunction at a cellular and molecular level. And the current challenge is basically to identify the pathologies of unveiling the spermatogenesis and identify the role of various components of spermatozoa in early human development. And hence clinicians all around the world are still looking for that one elusive sperm function test that could be universally applied in a routine lab as part of routine analysis. The principle of sperm function test is to determine the fertilizing potential of the sperm, and all the available sperm function tests helps us to identify sperm dysfunction at any one or more of the steps leading to sperm maturation and fertilization that occur during the passage of sperm in the female genital tract. Like the capacitation or acrosome reaction or sperm zona pellucida interaction, sperm oocyte membrane fusion, decondensation, and fusion of the male and female pronucleus. So based on that, they have divided the sperm function tests broadly into three categories as routine tests, extended tests, and advanced tests. In routine tests, we routinely perform sperm vitality and the Horst test. And in extended tests include the Teratozoospermia index, sperm DNA fragmentation, sperm aneuploidy, immunological tests like MAR and immunobead tests, and biochemical assays. And advanced tests includes the ROS testing, assessment of acrosome reaction, assessment of sperm chromatin, and CASA, etc. So moving on to the topic of today, that's I will be discussing the clinical relevance of most of the commonly used sperm function tests, and for which the demonstrations will be discussed by my colleagues in the coming forward. So we all know that ICSI is extraordinary, yeah. So in evidence over the last 25 years also indicates that the health of the ICSI offspring might be affected. Especially when the subfertility is of male origin. So the health issues potentially related to male subfertility and the use of ICSI could include congenital malformations, childhood cancer, psychological, neurological development abnormalities, infertility, and cardiometabolic profile impairment. So knowing all this, do we still want to ignore the functional competence of sperm before doing ICSI is the big question. So the first test is the sperm vitality. So this is routinely done as part of our semen analysis in andrology labs, and it is specially recommended when the progressive motility is lower than 40%. Clinically, it helps to identify necrozoospermia and points out to a number of potentially correctable clinical conditions that may contribute to impaired fertility. When all spermatozoa are immotile, it helps to distinguish between immotile, live immotile versus dead immotile spermatozoa, which again is critical in determining whether ejaculated or testicular spermatozoa should be used. Then moving on to the clinical relevance of the most commonly done sperm DNA fragmentation test. There was a survey conducted by Dr. Ashok Agarwal and group in 2017 that showed that sperm DNA fragmentation tests are commonly requested by fertility specialists. And they had prescribed tunnel assay, SC assay, and SCD and COMET assay commonly. And the top three indications for these tests were pregnancy failure after IUI or IVF for ICSI, and recurrent miscarriage after IUI, IVF for ICSI, and also recurrent natural pregnancy loss. The survey results showed that 2 out of 3 responders said that abnormal sperm DNA fragmentation levels would affect their clinical management. And so this paper also concluded that there is a well-established association between sperm DNA damage and infertility. And sperm DNA fragmentation negatively affects IUI and ART outcomes, but sperm genetic defect could be transferred via ART and could affect the health of the resulting offspring. A high DNA fragmentation result calls for action, which includes treatment of underlying correctable conditions associated with sperm DNA fragmentation, like varicocele, lifestyle factors, genital infections, etc. When no treatable condition is identified, consideration to ICSI with testicular sperm should be given, but the risk associated with sperm retrieval and ICSI with testicular sperm should be reserved for men with confirmed sperm DNA damage or severe oligoasthenoteratozoospermia or cryptozoospermia. So this is one of the reasons why testing is all the more important. Literature review suggests that there is a direct relationship observed between sperm DNA fragmentation level and poor IUI, IVF, and ICSI outcomes. So there's a study by Duran et al. That evaluated the outcome of 154 IUI cycles, and they detected significantly higher sperm DNA fragmentation levels among patients with failed cycles. And there's another study that reported that a lower pregnancy rate and live birth rate, like 3% versus 23% and 1% versus 19% live birth rate was found among patients with high versus low as sperm DNA fragmentation levels in IUI cycles. In IVF and ICSI, the current evidence features a significantly negative impact for sperm DNA fragmentation, but high sperm DNA fragmentation were associated with increased risk of pregnancy loss following both IVF and ICSI. Moving on to the next test, the genomic tests like sperm aneuploidy tests. They require experience and skill to increase its facility and validity. The clinical application of FISH is low because of the high cost and the technician timing and limited number of chromosomes studied in each round. FISH can detect the rate of aneuploidy in different samples, including ejaculated testicular and epidermal sperm. Clinically, results from this tool can be used in genetic counseling of couples suffering from male factor infertility or repeated pregnancy loss or implantation failure, and to make an informed decision. But routinely, we're not doing this on a large scale. The next is the most commonly spoken about ROS testing. So we all know that under physiological circumstances, the active observation species is highly necessary for optimal sperm function. They can potentiate sperm fertilizing capabilities like capacitation, hyperactivation, activation reaction, and oocyte fusion. But excessive levels of ROS that is produced by a variety of environmental toxins or pathological processes are detrimental. And it does result in, it does affect the health overall and fertility in particular. So to minimize these hazardous effects of oxidative stress, a number of endogenous enzymatic and non-enzymatic antioxidants also exist to scavenge and neutralize the excess ROS. Oxidative stress is present anywhere from 30 to 80% of infertile men as a result of generation of ROS from contaminating leukocytes, defective sperm, and antioxidant depletion. There are several methods for testing has been described, but their widespread use in clinical practice is hindered by their complexity and the lack of standardized techniques. But commonly, oxidative reduction potential measurement, ORP measurement, and chemiluminescence assay seem to be promising techniques that is capable of overcoming these obstacles. But further research is required to determine cost-effective, simple, and accurate technique that can be routinely used. The next one is the hyaluron binding assay. It has been developed as a commercial diagnostic kit for assessing sperm maturity. It is based on reports of hyaluronic acid selectively binding to mature sperm with intact acrosome and better morphology. So there's no clear threshold value that can be used for predicting sperm fertilizing ability, and clinical value of hyaluronic binding assay for diagnosis and management of male infertility is quite limited. And then anti-sperm antibody assay. So the clinical relevance here again, it's now quite limited because anti-sperm antibodies are present in almost 34 to 74% of vasectomized men and persist in about 38 to 60% after vasectomy reversal. So most clinicians still do not test anti-sperm antibody routinely because of their uncertain significance. And management options include corticosteroid therapy in cyclic doses and to increase antibody-free spermatozoa and selection of ICSI over IVF, IUI or IVF. CASA, again, the automation, it is potential as an advanced research tool, and not many centers have CASA systems and again, not used because of their cost and ease of use and everything. So CASA system needs to be rigorously tested and conformed to the differing needs of human clinical andrology labs. And further future CASA systems could maybe incorporate molecular biological aspects of sperm analysis into their database with the specialized statistical analytical capabilities. Having said all this, still we can, there are few sperm selection methods that could be practically used for ICSI rather than using the conventional swim-up or density gradient methods. And these are all based on other sperm function tests. So we have the host test that could be used or done before for selecting sperm for ICSI. Microfluidics and magnetic assorted cell sorting could be used in samples with high sperm DNA fragmentation. And we have PIXI and FermSlow, which are based on the hyaluron binding assay. And if none of this could work, we can go to testicular sperm as a last option. All these methods have their own pros and cons, and it's been debated enough. So the clinics can choose which works best for them, and there are numerous publications and studies on all of these methods. So emerging technologies are microarrays, proteomics, and metabolomics. Microarrays, they compare the transcriptomes at different stages of spermatogenesis and gives clues to the molecular mechanisms to genetic infertility and helps us to identify potential biochemical markers. Proteomics, because the seminal fluid has almost 923 proteins and with 20 plus proteins with altered expressions in infertile men. It is a non-invasive technique and helps to detect, it has a potential to detect various causes of infertility. So metabolomics, the byproducts of oxidative stress and naturally occurring antioxidants, they can serve as biomarkers to potentially differentiate fertile men from subfertile men. And the same principle can be applied to ART, as oxidative stress can affect pregnancy outcomes markedly. It is non-invasive and could be developed for use in clinical settings. So to conclude, it is very easy to criticize the sperm function tests based on the grounds of their low predictive values and variable thresholds. But we need to really rethink whether we are providing the best care to our patients and the child that is yet to be born by ignoring the health of the sperm. And there is still high quality clinical data missing on sperm function tests, and multicentric trials are essential to develop robust thresholds. new tools which are simple, cheap, reliable, repeatable, and effective, combined with more robust assessments are urgently required. So the primary objective is to basically improve our outcomes, but more importantly, to improve the health of the father and the resulting offspring. These are some of the references that I've used for this talk. Thank you very much, iHera team, for giving me this opportunity.
Thank you, Amita. Such an excellent presentation, and you touch all the sperm function test very precisely. And I think there is no question at all. But
there is a question.
I know that question, but it is not relevant to Amita. So we will take it after. That there is one topic as the that time we will take.
So in that case, I have one question for Amita.
Yeah, I have one question actually. That is the thing. So there are so many sperm function tests are there, but I don't think so. All the people are doing this. So just I want to ask you, which sperm function test you are doing in your lab?
We do sperm DNA fragmentation with the commercially available sperm chromatin dispersion assay. We have done quite a lot of study, even 3-4 years back, where we, for patients with high DFI, we have tried doing PIXI, microfluidics, and even sperm slow, and even testicular samples were used, and we have compared the outcomes with the regular density gradient methods. So when we have had, say, 15 oocytes, we injected 8 eggs with sperm with density gradient, and when we split the oocytes and we injected the rest with the sperm selected with any one of these methods, by microfluidics or PIXI, or very rarely we've done a few with testicular sperm also. So frankly, we didn't find major difference in our pregnancy results, but maybe it was not significant, maybe just 2-3% more, but not very significant. So we couldn't do anything more with that. So due to practical purposes, we moved back to our density gradient methods only, but still we do sperm DNA fragmentation method only.
So do you think, so that study which you have done in past, so there was large number of patients or there is small number of patients you have done?
No, we did around, I actually presented that paper in ACE 2017 in Hyderabad. It was around 60-70 patients, not much actually, I wouldn't say. And the testicular sperm volume was very low, less than 10, I think. So obviously our sample size was low. So, but we just tried to see whether it really made an impact. And this was done only for patients with high DNA fragmentation, not for the regular ones. We took the normal DNA fragmentation results as the control group and did that study.
So how do you decide that you have to go for that DNA? That what is the threshold you take?
That was patients with repeated IUI failures or repeated IVF failures, and if there was no particular female factor or some unexplained reasons, we didn't know where it was going wrong, then before going on to a next fresh IVF cycle, we did a sperm DNA fragmentation for the male partner.
Yes.
And 1 day of abstinence.
Yeah, yes, sir. Yeah, Amita, you covered everything. It was an excellent nutshell, but I have one question for you, which you missed out, I think. I want you to comment on the test with respect to rheotaxis or chemotaxis, or maybe also thermotaxis.
No, I have not done anything much on it.
Huh?
No, I have not done anything much on it.
No, I'm asking you, do you have any data on that? Like, do we have it, or how is it?
I have not had very particular data to share in this place. Yeah.
Fine.
I have done some experiments for thermotaxis.
Okay.
But the data was not that much conclusive. But I would love to do it now if we can, I think, combine something, and we'll talk on it. Yep.
Definitely, we can do that.
That I need more again.
Thermo, what temperature you have done?
Like you. Yeah.
Okay, thermo, what temperature you have done?
I have done 3 different temperatures. One is 38, 39, and 37.5. So basically 37.5, 30, and 39. And other side, the gradient was kept at on 36.
Okay, great.
Well, we'll connect on this. Yeah.
Yes, definitely. Well, sir, so thank you so much, Amita, for that excellent presentation. Again, thank you so much, Ved sir, for that moderation. And taking forward the session, I'll be introducing the next speaker. So I'll be introducing the next speaker, Tejshuni Sanket. She'll be talking on sperm vitality. She's currently working as a clinical embryologist at the Department of Reproductive Medicine, NFI Fertility Center, NFI Medical College Hospital, Mangalore. And she has pursued a master's degree in genetics from the University of Mysore. And she's pursuing a PhD in neurosciences from Mangalore University. And she's a team member of IHERA. And she's a member of ACE, ISAD, and Indian Science Congress.
Good evening, all. Thank you, Sanket, for the kind introduction. And today I'll be talking about sperm vitality test. And to introduce to my topic, the sperm vitality has estimated by assessing the membrane. Sperm vitality is estimated by assessing the membrane integrity of the cell, and it may be determined routinely on all samples, but is especially important for samples with less than about 40% progressively motile spermatozoa. And this test is recommended when total sperm motility is below 40%. The sperm vitality should be assessed as soon as possible after liquefaction of the semen sample, preferably at 30 to 60 minutes of ejaculation, to prevent the observation of deleterious effects of dehydration or of changes in temperature on vitality. And the percentage of live spermatozoa is assessed by dye exclusion or by hypertonic swelling method. The dye exclusion method is based on the principle that that damaged plasma membrane where the dye penetrates and stain non-viable, that is dead sperm, whereas live sperm resists the absorption of dye. And the hypoosmotic swelling test, it presumes that only cells with intact membranes, that is live cells, will swell in hypertonic solution. And coming to the methods of vitality assessments, the gold standard techniques are eosin-nigrosin staining method and hypoosmotic swelling test, which is also called as HOST. And both of these tests can be performed either by in-house prepared reagents or by commercially available kits. And in addition to this technique, there is a technique where the laser is used to detect the viable but immotile spermatozoa. Then coming to the vitality test using eosin-nigrosin stain. This is one-step staining technique which uses nigrosin to increase the contrast between the background and sperm head, which makes them easier to discern. And this is the list of reagent and the stepwise protocol to prepare the in-house reagents. And I'll be talking about the procedure. with this video. Firstly, mix the semen sample well. And it is checked for liquefaction. And once the sample is liquefied, (50 microliter aliquot of semen sample is taken in the test tube. Or you can also use the porcelain spot plate well for this procedure. And to this sample, equal volume, that is 50 microliter of eosin nigrosin stain is added. And mixed well. And it is kept aside for 30 seconds. Label the slide. And now 10 microliters of the suspension is taken and is placed on the 3/4 position of the slide, and by feathering technique, make a thin smear and allow it for air drying. After air drying, the slide is examined in bright field objective under oil immersion, and this is how the slide looks. This is how it looks under the microscope. And these white or transparent sperms, these are the live cells, and the head which are stained pink, these spermatozoas, these are the dead ones, because they take up the stain due to the membrane disintegration. Usually there are 3 categories of spermatozoa. The first one is unstained sperm, which looks transparent or white. There are live ones. And the second is leaky sperm, where the stain is taken up partially, and they can also be considered as alive sperms. The third one is the stained sperms, which take up the stain, and those are dead spermatozoas. Coming to the vitality test using eosin alone, this method is simple and rapid, but it is a wet preparation, and that's why it cannot be stored for quality control purposes. And these are the reagents used to conduct this procedure. And this is the procedure if you are using eosin only stain test. Moving on to the scoring, the live spermatozoa have white head, and dead spermatozoa have heads that are stained red or dark pink, and spermatozoa with the faint pink heads are assessed as alive ones. If the stain is limited to only a part of neck region and the rest of the head area is unstained, this is considered a leaky neck membrane. Is not a sign of a cell death and total membrane disintegration. These cells should be assessed as alive. The nigrosin provides a dark background that makes it easier to discern faintly stained spermatozoa. And moving on to the hypoosmotic swelling test. In HOS test, When live cells are placed in hypoosmotic media, the water enters into the cytoplasm, causing the cell to swell, particularly the tail. This assay does not damage or kill the sperm and is very useful for identifying viable non-motile sperms for ICSI. HOS has a limited ability to predict male infertility, but an HOS result, which is less than 50%, is associated with increased miscarriage rates. And this is the list of reagents and chemicals that are needed for HOS test. And then this is the procedure to perform the HOS test, and I'll explain the same along with the video. Before performing the HOS test, the HOS solution to be pre-warmed. One ml of HOS solution is taken in small round bottom tube. And is pre-warmed at 37 degrees Celsius for 20 minutes. After 20 minutes, the sample is checked for liquefaction. And 100 microliters of liquefied semen sample is added to the pre-warmed HOS solution. And it is mixed well. Then it is again incubated for 10 minutes at 37 degrees Celsius. After incubation, 10 microliters of the suspension is placed on a clean glass slide and is covered with cover slip. And then this preparation is observed under microscope. And this is how the sperms looks under the microscope. The coiling of tail can be seen distinctly. And there are different variations of sperm tail swellings where you can see the tiny bulge at the tip of the tail to the bulge which occupies the entire tail region. And coming to the scoring, swollen spermatozoa are identified by changes in the shape of the cell, as indicated by coiling of the tail. Live cells are distinguished by evidence of swelling of the sperm tail. Score all forms of swollen tails has live spermatozoa. And coming to the lower reference limit, the lower reference limit of vitality is 58% according to WHO 2010, whereas it is 54% according to WHO 2021. And the sperm vitality tests are useful in primary ciliary dyskinesia, where ultrastructural defects in sperm flagella results in absent or extremely low motility, but those spermatozoas will be highly viable. And also it is used to select sperm for intracytoplasmic sperm injection in surgically retrieved testicular tissue. Sperms are alive but generally non-motile because of lack of epididymal transit. And this article, which is published recently, showed that laser assisted selection of immotile spermatozoa has no effect on obstetric and neonatal outcomes of TISA-ICSI pregnancies. Coming to the take-home message, sperm vitality is a reflection of proportion of live membrane intact spermatozoa determined by either dye-exclusion or osmoregulatory capacity under hypoosmotic conditions. And the sperm vitality test, it can differentiate between necrosospermia and total absence of motility, indicative of structural defects in the flagellum. And accurate motility evaluation can also be verified by using sperm vitality tests, as the percentage of immotile spermatozoa should be higher than that of the percentage of dead spermatozoa. Vitality tests either by eosin, nigrosin, or host is simple, rapid, and it is also highly prognostic value, and it also has and it is also cost effective. Thank you.
Thank you so much, Tejaswini, for that wonderful presentation. So moving forward, we'll take up the questions later. We'll be going for the next lecture, which is about sperm DNA fragmentation. So we have Madhumanti for that, and due to some emergency, she was not able to attend live. So she has sent a recorded version of the same. So I'll be playing that.
Hello, this is Madhumanti Chakravarti. I'm working as an embryologist since 4 years.
Is it audible?
Yes, it is.
Okay.
In New Life Fertility Center, Siliguri, West Bengal. Today, my topic is sperm DNA fragmentation. I'll perform the test. You'll see how I'll perform the test. And thanks to iHera for giving me the opportunity. Special thanks to Dr. Sankit Dumal sir, who has chosen me for the discussion. So let's begin. Thank you. Sperm chromatin dispersion test is assessing human sperm DNA fragmentation. The integrity of the sperm DNA is being recognized as an important factor for successful reproductive outcomes. Sperm DNA fragmentation testing has gained importance as it offers a better diagnostic and prognostic potential than routine semen analysis. Several techniques exist to detect sperm DNA fragmentation, but sperm chromatin dispersion (SCD) test is a simple, reliable, and reproducible technique. Using SCD test, DNA fragmentation can be accurately measured using conventional bright microscope. So the kit contains, we are using sperm chromatin kit. Assess the semen sample for its concentration. Dilute the semen sample with culture media to 5 to 10 million per ml. Melt the agarose gel at 90 degrees Celsius by keeping it in the water bath for 5 minutes. Transfer the agarose gel to the heating block that is maintained 37 degrees Celsius and wait for 5 minutes. Then add 25 microliter of the sperm sample to agarose gel and mix it well. Place the sperm cell suspension immediately onto the pre-treated slides and place the cover slip. Avoid formation of air bubbles. Leave the slides at 4 degrees Celsius for 5 minutes. After 5 minutes, remove the slide. Carefully remove the cover glass by sliding it off gently. Take care to maintain the slide in a horizontal position throughout the process. Incubate the slide horizontally in solution A for 7 minutes. After 7 minutes, incubate the slide horizontally in lysis solution for 25 minutes, which is solution B. Leave the slides in distilled water for 5 minutes. Place the slide in 70% ethanol for 2 minutes, followed by 90% ethanol in 2 minutes for 2 minutes, and finally in 100% for 2 minutes. Next, leave it to dry at room temperature. Mix the solution C and D 1:1 ratio. Deposit a layer of the mix the solution C and D 1:1 ratio and deposit a layer of stain horizontally. Leave to stain for 15 to 20 minutes. Next, decant the stain and gently wash with distilled water and dry at room temperature. Next, visualize the slide under bright field microscope using 20x or 40x objective and grade the spermatozoa. The slide can be restrained if the staining are weak.
So basically solution A and solution B are nothing but your acid denaturant as well as lysis solution. So preparing the spermatozoa to take up the stain if there is any fragmentation. So it varies from company to company. Before performing any DNA fragmentation, you need to go through the user manuals properly because it differs from company to company. So she has performed the acid denaturation as well as lysis solution exposure. Now she also done denaturation she has treated with alcohol, 80-90 as well as 100% alcohol for 2 minutes each. Now she is preparing a working stain from the stock stain. So this will be your final stain where you're going to expose your fixed sperms. So I'll got the stain and place it on the slide where it covers the entire circle or the entire area where you spread your suspension. leave it for some time as prescribed by the company. Then decant the excess stain. Then you have to wash it under gentle running tap water, or you can also use a coupling jar where you can fill it up with the water, and you can remove the excess stain by dipping multiple times. So once this is done, allow it to air dry at room temperature.
Visualize the slide under bright field microscope using 20x or 40x objective and grade the spermatozoa. The slide can be restrained if the stainings are weak. Sperms without DNA fragmentation will be like big halo, medium halo, and sperm with DNA fragmentation, small halo, without halo, with halo, and
yeah. So once it is done, you have to score it under the microscope with the 40x mostly, and you have to look for the halos, number of halos, and there'll be big halos as well as medium as well as no halos. So those with a bigger halos were considered as the good sperms, and those with medium as also considered as spermatozoa, which are good in, but there is certain DNA fragmentation in them. And those sperms without fragmentation, without halos, are considered as the sperms with high fragmentation levels. And there will be a formula where how you're going to calculate the percentage of DNA fragmentation or DNA fragmentation index, where you'll be calculating like total number of fragmented sperms into 100 divided by total number of sperms that are scored. So that will be your the final percentage of DFI. So thank you so much, Madhu, for that neat presentation.
Okay, there is one question in that on DNA fragmentation.
Okay, sir.
So how much of a sperm concentration is required if we can do with 5 million per ml concentration?
So usually the company recommends the concentration is very high. See, there are multiple companies where they have different protocols. Most of the companies which has employed halo test kit or halo test method or sperm chromatin dispersion test, they recommend us to make up the concentration to 10 million per ml standard. This is what generally I've noticed. And if the concentration is too low, still we can go for DNA fragmentation. Only thing is make sure that you score at least 100 to 200 spermatozoa. In that scenario, you have to make replicates. That will help you out.
Can we do that in the scenario of low sperm count? Suppose there is less than 5 million or 2-3 million, but volume is good.
You
so
can concentrate.
No?
Yes, you can concentrate.
That is the question.
Yes,
and one more thing that from Teju's slide, it was very good actually. And I want to comment on that. She showed us both the things, vitality stain and host test. And on that, I want to I want to tell you, and I want to ask you rather than tell you that can you please tell our delegates about the host test, how we can choose this from a host, because she showed us that host solution is there. Right. Can we use that host solution in that particular for sperm selection?
Yes, sir. Usually what we do in case of ICSI is we streak the suspension from the host directly on the ICSI dish. Once you allow it for incubation, proper incubation along with the media, hypotonic media, then make a streak of the same on the ICSI slide, then start collecting the spermatozoa. Like you'll be having a pool of spermatozoa. Just you have to cross check with your number of harvest, number of oocytes that are lying with us to inject. So that is how we go with it, sir. Like aspirate as much as possible. Always say for assumption, for example, if you have 10 oocytes which are lying down to inject, then at least go for a collection of 15 to 20. Sometimes when you before injecting directly, you have to wash it with the PVP.
Yes.
You have to take it. You have to take them into the PVP drop, then wash it, immobilize. Then, of course, they'll not move most of the times, but still there are. We do get to some spermatozoa, even with the swelling also, they try to move. They show very twitching movements and all. So that is to go about it, sir.
Thank you.
And also I would like to mention one more point where she presented one of the paper where they used laser, low power laser to give a shot. Maybe I don't remember the power, maybe around 100-120 microjoules of power. And they just give a slight shot on the tip of the tail. And if there is any curling, then those sperms can be considered as live and they can be picked up for ICSI.
Right.
And even this data, and currently there is a clinical trial running on this, and they started last year. So the results are awaited.
So you know that I think this is very good. Actually, you know that sometime what happened, we are going to start the ICSI and we don't have any, you know, a sperm activator with us. So we can use a host at that time, host solution.
Yeah, host or laser, we can use it.
Yeah, sir. If it is laser is not also there, then easily we can use the host. So can you please tell one more thing? How to prepare the host solution if there is no host solution from companies available with us?
Sir, it is nothing. Yeah, nothing.
Yeah, that I know, but for delegates, I want to tell.
For the newcomers, this is a tip and trick where what we usually do when there is no host solution available, we'll be mixing the buffer. Most of the times, HEPs or MOPs with the ratio, like say, 100 microliters of HEPs with 200 or 300 microliters of distilled water. So that will become hypotonic solution, and expose your sperms in that for at least 5 to 10 minutes. Then definitely you're going to get the curls.
Yeah, that.
So this is a very quick and what we call jugaad in our language.
Indian jugaad.
Yeah. So that is what we do, sir.
Good. Thank you so much.
Thank you so much, sir. Next, I would like to introduce. Thank you so much, Tejashwini and Madhumanti. Next, I would like to introduce. Sorry. Not able to, yeah. I would like to introduce the next moderator, Dr. Paresh, who is a good friend of mine and currently working as a Chief Embryologist at Wings Group of Hospital, Ahmedabad. Dr. Paresh, please introduce the next speaker.
Yeah, thank you, Dr. Satya. Actually, sorry, my webcam is not working.
That's fine.
Thank
I'll know you.
Yeah. So our next speaker is Dr. Nidhi Singh. She is going to speak on oxidative stress in sperms. She is MBBS MD in anatomy, DNB anatomy, and at present she is working as a consultant and clinical embryologist. She worked at Vardhaman Mahavir Medical College and Safdarjung Hospital, New Delhi. She also worked at Center of IVF and Human Reproduction, Sir Gangaram Hospital, New Delhi, and IVF in Reproductive Biology Center, Maulana Azad Medical College, and LNJP Hospital, New Delhi. Dr. Nidhi, over to you.
Yeah, good evening to all. I'll just share my screen. Is it visible?
Yeah, it's visible.
Good evening to all. I'll be talking about on oxidative stress. So the topics will be discussed under the heading of introduction, mechanism of action, causes and effect, methods of measurement, prevention and treatment, and conclusion. So to begin with, everything in nature is about balance, and balance is needed for survival, whether it is human being or a single cell like the sperm. And sperm is the male gamete cell which undergoes various challenges along the way to form a gamete. Now, let's talk directly about the reactive oxygen species and how they affect the sperm. They are the unavoidable byproducts obtained from oxygen and metabolism. They are intracellular ROS concentrations determined by two things. The balances between, first is the rates of ROS production, and second is the rate of clearance by various antioxidant defense mechanisms. Now, at normal physiological level, these ROS are again, these reactive oxygen species are again very important. So even though they are toxic metabolites, they exert beneficial effects through regulating vital cell signaling cascades like the sperm maturation, including spermatogenesis, chromatin condensation, spermatozoa maturation, and epididymis, the hyperactivation of the sperm, capacitation, acrosomal reaction, and finally in the fertilization. Now, the ROS concentration, when exceeds beyond the physiological limit, it can be because of two main causes. Either the ROS generation overwhelms the antioxidant threshold for the clearance, and when the antioxidant production itself is diminished. So what it leads to? It leads to oxidative stress, and this is the thing which we'll be discussing. So it's an imbalance in redox potential, which carries significant negative effect on various cellular components like lipids, acids, proteins, etc. Now, why sperm? The question comes in our mind. It is because there's inadequate cell repair system. There is insufficient antioxidant defenses because of the very little cytoplasmic content present in the sperm. It is susceptible to lipid peroxidation because of high content of polyunsaturated fatty acids in the plasma membrane. Therefore, there is disruption of the membrane permeability, leading to efflux of ATP and ultimately leading to impairing of flagellar movements. So these are the outcomes leading defect in the male sperm, causing motility problems, sperm viability problem, and fertilization potential, thus causing male infertility. Now, this is a very beautiful diagram taken from the article Oxidative Stress in Varicose, which is depicting the ROS and the antioxidant balance. I'll be jumping forward. We have already discussed most of it. And the ultimate cellular destruction which happens is the mitochondrial dysfunctioning due to the oxidative stress, protein denaturation, sperm apoptosis, lipid peroxidation, and finally mitochondrial and nuclear DNA oxidative damage. Now, this is at the cellular level, and it has a lot of cell cycles and a lot of factors involving, which is beyond the coverage of this lecture. So we'll come to the measurement of the oxidative stress directly. So literature states more than 30 different assays for measuring seminal oxidative stress. Now, there are direct tests which measures the degree of oxidation within the sperm cell membrane, and there are indirect tests estimating detrimental effects of oxidative stress, such as DNA damage, etc. Just a minute. Or lipid peroxidase levels. Now, this is an article in which all the tests are discussed, but I'll be mainly concentrating on these 5 tests which are commonly done on routine days. Now, coming to the first test, that is a routine semen analysis. What are things which we observe is, first are the semen parameters, where we see the sperm count, morphology, and motility, which is directly in conclusion to the oxidative stress. It is seen that acinospermia is perhaps the best marker for oxidative stress. Now, coming to the second test, which is hyperviscosity of seminal plasma. When it is present, it indicates that there is rise in seminal plasma MDA, and there is decrease in seminal plasma antioxidant status. Thus, it is measured by 4 grades, that is very low, low, high, and very high. It is seen by measuring the threading in the semen. The third method is the detection of round cells, that is leukocytospermia. Whenever there is leukocytospermia, it is directly in concentration or relation to exaggerated ROS production. So here we can see the round cells in this diagram. The round cells, but the point to keep in mind is these round cells should be differentiated with immature spermatozoa cell. So further special tests are done like the peroxidase test, which can discriminate between the two. Now coming to the fourth, that is disrupted sperm morphology and cytoplasmic droplets. When it is increased, it states there is presence of anomalous spermatozoa, again, which is seen in cases of uncontrolled ROS. And the fifth test, which we see most commonly, is the poor sperm membrane integrity. That is the host test, which has already been discussed previously, in which we can see the curling of the tape. Now coming to the most commonly used test, which is for the ROS, which is the chemiluminescence assay. Now what it requires is 2 main things, that is luminometer and a chemiluminescent probe, that is luminol. Now what is the procedure and how the steps goes around is, first we take an aliquot. Sorry, I could not provide you with the video. The aliquot of the liquefied semen is centrifuged at 300 G for 7 minutes. Then this aliquot is frozen at -20 degrees Celsius, so that the whole of the aliquot is used for the calculation of antioxidant levels. Now the pellet later on is washed in PBS, and 400 microliter of this aliquot is used and is suspended in the washing media. Now this is the machine which we use for chemiluminescence assay. Now if we see what happens, actually there are 4 tubes in which we are taking. First is a blank tube. The second, the blank tube here, in which only, excuse me, just a minute. Yeah, in which the 400 microliter of PBS solution is taken. Second tube is a negative control tube. Okay, where 400 microliter of luminol is added. The third tube is the test sample tube, in which again there is addition of the sperm suspension. Now what happens exactly? The luminol which is added to the mixture serves as a probe, and the test tubes are loaded into the luminometer later on for 15 minutes to measure the level of ROS. And the fourth test tube which we take is a positive control test, in which 50 microliter of H2O2 chemical is added. Now what happened when luminol measures both? First of all, luminol measures both extracellular as well as the intracellular ROS present in the semen plasma. Now free radicals are present in the semen sample. They produce light signals which react with the luminol. Now what happens? These light signals are converted by the luminometer, and then electrical signals of photons are released, which are read by the machine. The measurement of the number of free radicals generated is done as relative light units per second per million sperms. So what is the normal range of ROS level seen in a washed sperm suspension? It is stated as 0.10 to 1.03 into 10⁶ counted photons per minute per 20 million sperms. Now this is a very important parameter which we should remember. Now in routinely what happens? This chemiluminescence test is not done in a routine in everyday lab. So what normally we do, we have a kit provided by multiple companies. This is a picture which I've taken. In this routine kit, what we do, there is a dye known as nitro blue tetrazoleum dye, which interacts with the free radicals present in the semen sample, and they generate various amount of colors, starting from light pink to dark purple. Two important things are one tube is containing agarose and gel tubes are present, and second is a float which is used for heating of the tube. And this is a picture in which the different grades are seen, and we compare the tube after adding the chemical to it and keeping it for 15 minutes. Now coming to the next test, that is a TAC test, total antioxidant capacity test. So in this again, luminol is used for the measurement of TAC within the seminal plasma, which luminol is quantified against vitamin E analog that is known as Trolox. The principle is that there is reduction of copper 2 ions to copper 1 ions by the presence of antioxidants. So this is the kit again which is used by them. In this, the antioxidants is measured by absorption at 450 nanometer using colorimetric method. So how was the test interpreted? It is interpreted by using a Rostak score. It indicates that combined antioxidant activities evoked by all the constituents, that is whether they are vitamins, lipids, and proteins. Now this is the chart which is shown. This is the normal curve which is shown, and these are the curves when there is presence of multiple measurements of various antioxidants present in the semen sample. Coming to the next is a LPO marker, that is a lipid peroxidase test. Lipid peroxidase accumulates in the spermatozoa, producing a variety of decay end products measured as indicator of oxidative stress. It's an indirect test. Now they are the MDA, that is aldehyde, that is isoprostatanes, 2-propanol acrolein. Now the mechanism or method of choice is again a thiobarbituric acid assay. Now what happens? We add MDA to the thiobarbituric acid, and it produces a byproduct that is 1:2 adduct. And this byproduct which is produced, it produces a color which is measured by fluorometry or spectrometry. The next test is a seminal oxidative reduction potential test, also known as redox potential. It measures the potential of electrons to move from one chemical species to another. It measures the relationship between the antioxidants and the oxidants. Now how it is done? This is the device in which there is presence, there is a display screen is there. This is a sensor socket where this is injected. And the socket consists of the port where the sample is added, and the ORP is in semen is measured by using male infertility oxidative system. Now this is the graph which is produced comparing the normal semen with the normal semen parameters. And it is said that this test is standardized, reliable, and reproducible for research and investigation purposes. Now, what are the different studies and correlation? The studies done in infertile man versus fertile men, there are multiple studies. I'm just taking the zest that the median ROS level is 124 times higher in infertile male, percentage of sperm with cytoplasmic droplet significantly higher in infertile males, and there is higher percentage of spermatozoa with fragmented DNA, as we have seen and will be seeing later on in the other lectures also. Now, the negative effect of ROS with positive correlation between acrosomal anomalies and MDA values is seen in most of the tests. In infertile male, the oxidative reduction potential and the sperm DNA fragmentation shows significant positive results with sperm defects and significant negative correlation with normal sperm morphology. So what are coming to the last part, that is, what are the preventive measures and management of this male reproductive oxidative stress? Now, the first step is verifying the underlying cause or the causes. What can be there? What are the causes, as we have seen earlier? Then the management includes lifestyle modification, increased professional and personal stress should be counseled. The bad habits should be removed. Exposure to pollution, heat, toxin, and heavy metal, noxious chemicals and vapors should be ruled out. Activities raising the scrotum temperature like saunas, hot baths, long period of driving, and long sedentary office hours, along with also varicose, which leads to the increase in the scrotum temperature, should be seen. Now, in coming to treatment, the first line of treatment are the antioxidants, which eliminates the ROS and reduces their formation to halt the oxidative chain reaction. Now, again, they're divided into two main categories. One is preventive and preventive antioxidants, and second is the scavenging antioxidants, vitamin E and C. I'm just going forward. The second category of antioxidants, which we see, are enzymatic antioxidants and the non-enzymatic antioxidants. Non-enzymatic antioxidants are mainly which are present in the multivitamins which we eat nowadays. Now, there are many studies done, multiple studies done on the effect of these antioxidants. So they reported a significant reduction in oxidative stress or DNA damage after treatment with antioxidants in 19 out of 20 studies which they have selected. And again, I'll come to the zest that it is seen that this particular advantage of treatment of oxidative stress in cases of male infertility when we use multivitamins and antioxidants. Now, the last method of treatment is surgery, especially done in cases of varicocele. Varicocele is where the abnormal elongation or dilatation of the pampiniform plexus of veins takes place surrounding the spermatic cord, thus leading to the increase in the temperature of testis. The corrective strategy is known as varicocelectomy, in which these veins are being ligated, they are occluded, and thus in turn they reduce the seminal ROS levels, protecting the sperm from oxidative damage. Now, what do I conclude? I conclude that oxidative stress is directly related to increasing male infertility. Despite the growing body of evidence, oxidative stress testing is not routinely indicated for the evaluation of infertile male. The reason is mainly related to the availability of the test, the complexity of the test, the cost effectiveness, and most commonly, the lack of universally accepted test protocols. Thank you.
Thank you, Dr. Nidhi, for a nice presentation. And you have covered all the diagnostic and treatment for the antioxidant.
Thank you, sir.
I don't find any question answer. But actually, why do we worry about the oxidative stress? Because we know that when we are talking about the oxidative stress, it's a major cause of sperm DNA damage. And that DNA damage, which it lead to the recurrent spontaneous abortion, childhood cancer, genetic and epigenetic defect in offspring. So this is not a routine procedure. Routinely, we are not checking the oxidative stress. But I suggest whenever you find, as Dr. Nidhi said, there is a difference in motility and morphology, hyperviscosity. You found more amount of the round cell like leukocyte, and also you correlate with the higher fragmentation in day 3 embryos in the same patient. You can ask your clinician for the antioxidant treatment, because you know that when we give the antioxidant treatment, your study said 19 out of the 20 will reduce the oxidative stress and will give you will end up with a good result. So again, thank you, Dr. Nidhi, for nice presentation.
Thank you, sir.
Yeah,
moving forward,
I'll
just have a quick question. Yeah,
Yes,
Paresh, I'm allowed to take a question.
Yeah, sure, sir.
Yeah, Nidhi, it was good. And the thing was, see, you mentioned about liminometer, you mentioned about calorimeter and calorimetric estimation, as well as flow meter and all. See, when you practically look at it, the cost wise, the equipment and the quality control part, and establishing the SOPs for the same is quite expensive and also very laborious and is very intensive. So I just want you to comment quickly on when you correlate all these techniques with rapid kits, whatever available in the market.
Yes, sir.
What do you find? See, most of the times within 20 minutes, after 20 minutes, we'll start extracting the sperms from the plasma.
Yes, sir.
So we are almost reducing the burden of OS. But I just want the quick prognostic value when you compare this test with rapid quit.
So to be honest, I cannot comment on this since I have not done. That is why I was not able to provide with the video. I have not done this testing in routine, and I could only get the data from the different studies which were done. But practically speaking, what I have seen is, irrespective, we use the rapid kit, but the outcomes are not. You can say they are not. Good
Reproducible.
Reproducible. They're not reproducible because of the many factors like temperature changing, handling of the sperm sample, delay in the liquefaction of the sample. All these lead to the increase in the ROS. So they are not the right thing. That is why these assays are better, and actually they are more significant in finding out the defect rather than the rapid kits, sir.
One more thing in that, you know that in that rapid test, you can just do the qualitative, not the quantitative.
Quantitative.
This is the big issue in that.
That's the important thing. Yes, right. Maybe that is
and one more thing, that when you are doing DNA fragmentation, that I don't think this is recommended for that ROS is recommended. It is some like both are actually related to each other.
Indirect method of measuring.
Indirect, yeah. That is the thing.
Right.
But definitely in some cases when there is too much high ROS level is there, so definitely it is good to go for when there is nothing, you have find unexplained infertility is there, and patient is also going to select their own. They are going to doing IVF using their own gametes. Then it is better to go with this expensive test. Otherwise, I don't think there is any relevance of that.
So basically not for all, only
for
not
for all, not for all. And you know that I tried to buy that luminometer. It was cost around 4 lakh.
Very expensive, sir. So
and 4 lakh only 4 lakh only for that luminometer.
Sir, standardization is a lengthy procedure.
Yeah. And then running cost also, sir. Running
cost and everything.
The chemicals, so they have a lifespan.
The kit might cost you around 20,000, maybe one-time quit.
Antioxidant treatment is cheaper than that equipment, actually.
Yeah, you are right. You are right.
Yes,
I think one thing which is there is most commonly the test reproducibility, like has been said, it is not standardized because of multiple factors. And at the end of the day, you have highlighted that whatever they are reflecting in terms of better morphology might be better motility and that way. But how much of an advantage do these give when it comes to the pregnancy rates?
Prognostic
value. I think one of the main reasons, prognostic value. How much of the prognostic value, how much does it actually improve?
True.
I think one of the main reasons why it has not been taken up is it does not reflect in the pregnancy rates. The improvement in the pregnancy rates, what I feel it has not been taken up majorly.
Even that is what literature also tells. Data is very scarce.
Though it seems very good on paper, oxidative stress should have caused the problem. But when it comes actually to the practical things, might be standardization is lacking, or the selection of the patients whom it might be there, or the gradation is lacking. So probably
I think that is the main reason why WHO 6th edition has moved it to the research category rather than the basic examination. Dr. Paresh, please.
Can we introduce our next speaker? So our next topic on anti-sperm antibody. And I would like to introduce Nancy Sharma Brahmbhatt. She is working as a chief embryologist and lab manager at Ahmedabad, IKDRC. She is a ASRM certified embryologist and certified from ASRM Infertility Preservation for Reproductive Health Providers. Embryo and blastocyst biopsy training workshop from Embryology and PGD Academy, UCL. She has also taken embryology certificate course from Indian Fertility Society, and she has work experience more than 7 years in field of ART. She has submitted more than 13 research papers internationally as a presenting author, and she is PhD scholar and postgraduate in molecular biology and biochemistry, Guru Nanak Dev University. She is a member of prestigious national and international societies, ASRM, ASRE, AS, and IRA. Dr. Nancy, over to you.
Thank you, sir, for this kind introduction. My slides are visible to everyone?
No, it's not visible. Just do the slide so. Share,
Is it visible?
share your screen.
No, share screen karna.
Share screen.
Now?
No.
Or now?
No.
I have shared my screen.
No, Nancy.
No, it's not visible.
You should
Still not
do
visible?
it. Do it again. Do it again. Just cancel it and do it again.
Okay, sir.
Yeah, now it's visible.
Okay, sir.
Yes. Full screen.
Full screen it is?
Yes.
So, good evening, everyone. Today I'll be discussing on anti-sperm antibodies. So here we are having our learning objectives. So at the conclusion of this presentation, we all should be able to know what is anti-sperm antibody, what is the mechanism of action behind it, what causes anti-sperm antibody production, what are the implications on fertility, what are the diagnostic tests available, and what treatments we can offer to patients. So let us start with what is anti-sperm antibody. So like the name says, anti-sperm antibody or autoantibodies fight against sperm. It happens when the immune system mistakenly targets sperm as an invader. Both men and women produce anti-sperm antibodies, and this kind of infertility is usually called as immunological infertility. ASAs are immunoglobulins which are directed against the antigens which are present on the sperm surface. So we are having IgG, IgA, IgM. And if we talk about IgG and IgA, they are frequently found in semen. IgG occurs in genital tract fluid and is either produced locally or transduced from blood serum. IgA is a secretory type and always produced locally. Now, IgG and IgA, they both have negative impact on sperm motility, while IgM is present only in blood serum, and it is basically present during the acute phase of infection. Now, moving towards the mechanism of action, how anti-sperm antibody produces, and what are the reaction of anti-sperm antibody when it interacts. So mature spermatozoa are normally located behind the blood-testis barrier. Here we are having the blood-testis barrier in the picture, and all the mature spermatozoa are behind this barrier. So Sertoli cells are responsible for the formation of blood-testis barrier with their tight intracellular junction that divided the seminiferous tubules into two seminiferous epithelium into the two compartments. So what happens, spermatozoa are physiologically always unexposed to the male immune system. However, when the blood-testis barrier is broken or breached here, like this, here, when it is broken and breached, anti-sperm antibody produces. Or we can say that the immune system leads to development of anti-sperm antibody. Although not all anti-sperm antibody impairs sperm function, but anti-sperm antibody may alter motility, acrosomal reaction, capacitation, fertilizing abilities, and other things. So now moving towards the causes of anti-sperm antibody. What causes anti-sperm antibody? So autoimmune reactions against spermatozoa can occur after testicular epithelial vascular injuries that can lead to the exposure of sperm to immune system, surgical traumas, testicular cancer, undescended testes, urogenital inflammatory condition, obstructions, and varicocele. All these have been associated with the presence of anti-sperm antibody. So ASA is strongly associated with obstructive azoospermia, particularly post-vasectomy. Post-vasectomy, we have 70 to 100% of men having the presence of anti-sperm antibody. So now, what are the other factors which affect or which lead to the production of anti-sperm antibody? Here we are having chronic bacterial infections and prostatitis also, that also accompanied anti-sperm antibody, and that could be associated with the inflammatory damages to male genital tract as well as local immune dysregulation. That can lead to autoimmunity against the spermatozoa. Now, it has been also suggested that ASA could be produced during cross-immune reaction with exogenous antigens like bacteria, viruses, fungi, or allergens. So there is one recent study that suggests that HPV virus is associated with the high risk of production with anti-sperm antibody, and that affects sperm quality, in particularly sperm motility count, and may play an important role in male infertility. Now, moving towards the what cause, we have already discussed what are the causes behind it. Now, how it affects fertility. So after the production of anti-sperm antibody, the anti-sperm antibodies are significantly associated with reduced sperm production, transportation, sperm motility. This is how it affects infertility via male. And if we talk about ASA circulation in serum, it can affect gamete interaction, fetal development, early embryonic development, implantation. There is also a few papers which suggest that it affects implantation, but the data is still insufficient for that. According to a previous meta-analysis, asthenozoospermia may be an indicator for the testing of ASA. Now, moving towards the tests which are available. So we are having agglutination test, complement dependent test, immunoglobulin binding test, mixed immunoglobulin reaction test, ELISA, PCT, and hemizona assay. So only two tests are recommended by WHO, that is IBT and MAR, that is immunoglobulin binding test or mixed immunoglobulin reaction test. So though the mere presence of sperm antibodies is insufficient for the diagnosis of sperm autoimmunity, it is necessary to demonstrate that the antibody interferes with the sperm function or not. So if it can interfere with zona binding, it can also interfere with the acrosomal reactions. So we'll be discussing about briefly about agglutination test, complement dependent test, ELISA, PCT, and HZA, and in detail about IBT and MAR. So agglutination test, if we talk about agglutination test, and sperms demonstrate agglutination when motile sperms stick to each other by head to head or tail to tail or in mixed way, the presence of antibody is one of the most possible cause of the investigation. But it should be noted that the sperm antibody can be present with sperm agglutination and equally without sperm agglutination, and it could not be, it is also one case when sperm antibody production is not associated with and agglutinations. So next we are having complement dependent test, or we can call it cytotoxic effect. As we know, when sperm is exposed to serum, they are getting killed by the immune system, and the presence of antibody exerting a cytotoxic effect might be suspect by reduced or declined motility and viability. So the disadvantage of this complement dependent test is that it cannot detect IgA class of antibody. Now moving towards ELISA. ELISA can be done to detect ASA. It is very specific and quantitative, but very time taking and costly, and also it is unable to determine where ASA location and the type of isotype we are having. Moving towards post-coital test, where we examine the cervical mucus several hours after the intercourse and the presence of sperm under the microscope. So this PCT and HZA, that is hemizona assay, can be also done in association with the IBT test, both directly or indirectly. And this we will be discussing further in the presentation. So the most important thing here is to notice that there is low prevalence of this. 2 to 4% of ASA has prompted by ASRM, American Urology Association, European Urology Association, and NICE guidelines says that it is not endorsed that ASA tests are as first line test for evaluation of infertile men. Now, moving towards the two main categories which are given by WHO, two main tests which are provided or recommended by WHO. We are having MAR, mixed immunoglobulin reaction test, and immunobead test that can be done directly or indirectly. What is the meaning of direct and indirect? So directly is when the when we test for antibodies on spermatozoa, that is the direct test. And when we test for antibodies in sperm-free fluid. Sperm-free fluid, like when we tested in serum and follicular fluid and other things. So it is indirect. Now moving, it can be that I have already told that it can be done both directly or indirectly. So what is the direct test? We have to incubate sample with latex bead coated with anti-human antibodies. Then these anti-human antibodies on bead binds to the anti-sperm antibodies on sperm surface, and then we have to observe under the microscope for the results. It provides information about the presence of immunoglobulin that at specific location, that whether they are present on head, whether they are attached on midpiece or a tail or all of three. Now, the most important part here is it depends, the direct test is always depends upon the presence of motile sperm. In case of inadequate number of motile spermatozoa present, we can anyways go for indirect test. Now, this is a picture of how the binding at head, tail, and midpiece looks like. Now, we are discussing direct MAR test for IgG and IgA test. We can do it for IgG and IgA similarly. So, first of all, you have to take all the reagents to 37 degrees Celsius. Then you have to label the slide of patient with a particular patient ID. Then you have to take 5 microliter of raw liquefied semen sample to the slide. Then add latex coated particles of IgG to it and mix thoroughly. Then you have to again add 5 microliter of anti-sperm antibody to it, anti-serum antibody to it. Then mix thoroughly, make a wet preparation to it, add cover slip. Then you have to keep it for 3 minutes and in the petri dish for incubation. After 3 minutes, you can see it under the phase contrast microscope. Again, repeat the process by doing it for 10 minutes. And again, see the results under the phase controlled microscope in 40x. So we'll be discussing results after the indirect MAR test. So this is how a direct test happens directly with the semen. Now, how an indirect test happens? Again, for that, we also have to take all the reagents to room temperature, at room temperature. Now you have to label the tube with the patient ID. Then you have to take 50 microliter of serum and dilute it to 1:5. Then you can add 250 microliters of media to it. Again, label the tube. Then add 250 microliter media to it and mix thoroughly. Add semen sample to it, 50 microliters. Mix it, try avoiding the bubbles. Keep it for incubation for 37 degrees Celsius for 50 to 60 minutes. Then label the slide with the unique patient ID. Then again, the process is similar as direct. So then you have to take 5 microliter of serum, then latex coated 5 microliter of latex coated particles of IgG or IgA, whichever test you are doing. Mix it thoroughly, make a wet preparation to it. Incubate the slide for 3 minutes in a petri dish. Observe under the phase contrast microscope. Again, repeat the process by incubating it for 10 minutes. And again, see it under the phase contrast microscope in 40x. So how the results are, how do we have the results? So we have to evaluate 200 motile spermatozoa to achieve an acceptable low sampling error. So this is how it looks. If a sample is negative in result, it is not bounded to the beads. And if a sample is positive to the result, it is bounded with the beads to it. Now, MAR test is the most commercial test available. Now we are moving towards the immunobead test. Immunobeads are the plastic particles with the attached anti-human IgG antibodies. No commercial IB test is available. That means aminobeads must be made inside the immunological laboratory. CNBRs are activated support speed to conjugate anti-human IgA or IgG to the bead. It is more time consuming but provides information about antibodies on the sperm that have removed from possible masking component in the seminal plasma. We have to take both positive and negative controls for each other here. How do we do it? We have to place 5 microliter of washed semen sample. The major difference is you have to take washed semen sample for it. Also prepare a positive control and a negative control on different slides. Then add 5 microliter of anti-Ig immunobead suspension beside each sperm droplet and mix it. Then store the slide horizontally for 3 to 10 minutes, firstly for 3 minutes and then observe, and then for 10 minutes and then observe at room temperature in petri dish, and examine the slide on the phase contrast microscope at 400 m magnification. This is how it looks when you, this is the first test of example with negative, and this one is the positive aminobead test, where aminobeads adhere to the motile sperm that have surface bound antibodies, and if our text mixture is direct test is scored positive if we have at least 20% of the sperm cell binded to it. So this is a beautiful review article on diagnosis and treatment of immunological infertile men with anti-sperm antibody, which I have taken. Now moving towards the direct IBD, there is this in this paper, there is a beautiful approach to how do we do IUI and how can we plan ICSI for this kind of patients. So I'm just giving the overview of how it is done. So after direct IBD test, they have also included post-coital test and hemizona assay to it. If the post-coital and hemizona assay are normal, the patients have been counseled for timed intercourse. And if we are having abnormal PCT and normal hemizona assay, the patient is counseled for IUI. And if both are negative or both are abnormal, the patient is directly counseled for ICSI. So why we are doing hemizona assay in here? The normal hemizona assay where sperms are bounded to the zona pellucida. In this hemizona assay was carried out to evaluate the blocking effects of ASA on fertilization. This is how it looks in the first picture. There is positive hemizona, and in second picture there is negative hemizona. How do we calculate it? This is the formula for calculating it, which was given in the paper. Now moving towards the medical managements of infertility. How do you manage anti-sperm antibody? So WHO recommends that the diagnosis of immunological infertility should only be made after evaluation of the impact of fertility by ASA and if it affects sperm function. So we have two different approaches to it. We have corticosteroids and ART. So if we talk about corticosteroids, we are having here, if we are talking about corticosteroids, we are having 3 different treatments here. The decreased ASA production, which are systematic corticosteroid, it is relatively insufficient. And when we remove the sperm bounding after washing, it is also insufficient. From here, we can only see that the ICSI is the most excellent chance for anti-sperm antibodies. Now moving towards the corticosteroid administration, the oral corticosteroid used to suppress antibodies production and have been reported in relatively older literatures to have beneficial effects on the men with ASA. But there is this RCT that report that the corticosteroid administration in men with ASA could improve conventional IVF and outcome. However, the studies also suggest or says that ICSI outcomes were not improved by corticosteroid. That is because ICSI can bypass ICSI can definitely bypass the natural process of zona binding and fertilization. But the most important thing in the corticosteroid administration is that the immune suppressant therapy may have adverse effect, and it outweighed the potential benefit of steroid overtreatment that could result into the vesicular necrosis of femur head, hypogonadism, diabetic onset, metabolic decompensation, and male infertility. Now, the dose and the duration is very much important, and it should be carefully considered. So here I am happy. There are some references I have taken from over the 3 decades, which suggest that clinical utility of prescribing medical immunosuppressing therapy for the sole purpose of suppressing ASA is not clear. Now moving towards ART. So, firstly in ART, we can do semen washing that can remove unbounded antibodies to it. we can suggest patients to collect sample in in culture media, 1 to 2 ml of culture media to it. ASA and mainly bound to spermatozoa post-ejaculate, and this diluting effect could impede ASA binding to motile sperm, which can be separated by washing. So now, what is the golden standard for immune-mediated infertility? The technique for the golden standard is ICSI. So ICSI is the first choice as it yields to higher pregnancy rate. According to all the papers, it produced to higher pregnancy rate per cycle and as compared to IUI and IVF. So here are some references for IUI, IVF from the latest literature, IUI, IVF and ICSI. For IUI, here we are having a 2020 study which suggests that lower natural LBR in group 1, whereas 100% ASA positive patients, and comparable LBR are there with the IUI, having the patients with group of 50 to 99% of ASA positive patients. After this, we are having IVF. In IVF, we are having a 2019 Liu et al. Study, which suggests that with 39 ASA positive patients, there is lower rate of fertilization, but good embryo formation and PR and NPR in ASA positive than in ASA negative patients. For ICSI, we are having the similar study, which says that for both ASA positive and negative patient, when we do ICSI, there is comparable pregnancy rates and LBR rates. So now I just want to summarize with the few things that what the main indications of the test we have already seen, that these are the main indications of the test. The thing where I want to add here is asthenozoospermia, especially in case of if we see agglutination here, is an indication for the testing. If we talk about the methods of testing, definitely MAR and IB tests are there for method of testings, and indirect test detects antibodies that are found in free fluids like seminal plasma, cervical mucus, and can be performed in obstructive azoospermic patients and oligozoospermia, or if semen needs to be stored for the later testing, can be also done. And for management of immunological infertility, definitely ICSI can be overcome infertility due to ASA, and it is recommended ART treatment for it. And it is also noted that if a patient is scheduled for ICSI, there is no need for the test of ASA, as there will be no effect on the outcome. This is already given in all the literatures I have read. Thank you so much for your time and patience.
Thank you, Dr. Nancy, for covering all the aspect and beautiful presentation. Do you do any test at your center?
No, sir.
No, I'm just asking because you are in one government very good center. Fine. So actually, anti-sperm antibody, mostly the clinician, they are not prescribed this test, so we are not doing. But whenever we are checking the sperm agglutination affecting the motility, then we can go with the anti-sperm antibody. We know that in male infertility, there are around 5 to 15% of infertile men because of the anti-sperm antibody.
Yes.
Routinely, this antibody is not present. But what happens when there is a blood testis barrier is broken down, and due to high infection and blood mixed with this sperms, and there is a chance that the antigen which are stuck to the head of the on the sperm, so antibody will attach to the sperm, and again it affect the motility. So definitely the treatment are corticosteroid, sperm washing, and IUI, and very true that ICSI is a gold standard. And mostly I cannot say all of us, but most of the people, they are doing ICSI for all. So definitely here ICSI helps us for this anti-sperm antibody. So again, thank you, Dr. Nancy. We don't have any question and answer.
Just one.
Can I ask one question?
Yeah.
Yes, like has been said, acinospermia could be an indication. A good presentation, Dr. Nancy. Like you said, acinospermia is an indication for testing for anti-sperm antibodies. Can you tell us like what specific kind of acinospermia are you looking at?
So
what is it when you see under a slide?
If we have asthenozoospermia with agglutination, that is definitely an indication for ASA testing. And either if we have a partial agglutination test for it, we can take that patient of asthenozoospermia for ASA testing. This is what
Any pattern of acinospermia?
Sorry?
Any specific pattern you will see? when you should advise? See, acinospermia will be seen
No,
in
no,
so
sir.
many patients.
I'm saying if you see asthenospermia with agglutination. If
In
you see
your experience,
asthenospermia with
how many
agglutination,
would
it
you
is
have seen
already,
agglutination
sorry?
site-specific? Sorry, how many site-specific agglutinations have you seen? Like, how commonly do you see? See, for us to say agglutination, it has to be site-specific, right?
Sorry, sir. Yes.
But then if you are saying 5 to 10% of the cases, you are saying anti-sperm antibodies might be present, right? But
Yes.
my question was like, how many times have you seen in your practice site-specific agglutination under the microscope slide?
Site-specific agglutination. No, so the agglutination is definitely divided into different types, and when there is head-to-head attachment, tail-to-tail attachment. So I have seen head-to-head attachment mostly in few cases which I have seen. But if I haven't that, like that, I said that we are not doing assay testing for such kind of patient. If we have any kind of doubt that this patient is going to have anti-sperm antibody production, we will definitely take that patient directly to the ICSI. So that factors eliminate there. I have already said that we are not doing in routine, or we are not doing anti-sperm antibody test.
Yeah, actually, what like one of our persons has mentioned in the chat, I think what we usually look for is vibratory kind of motility will be seen. That's
So,
when you need to consider.
So that the one paper I have read, that post-coital and hemizona assay paper, I have seen that if there is 10 to 20% of motile or progressive sperm after post-coital test, so then that patient is normal. And if we have twitching or immobilized sperms, or we can say that just moving sperms in which are less than 10 to 20% after post-coital and hemizona test, that patient could be also taken for ASA testing.
Sorry to
also
interrupt.
one
Sorry
paper.
to interrupt. Nancy, you
Yes,
must remember in 6th edition, PCT as well as HZA, both have been discarded. So entire IBT will become no point now, right?
Yes.
Yeah.
I
So
feel
I have one
PCT is
question,
underutilized,
Nancy.
but
I have one
for
question for
another
Nancy.
discussion.
One simple question. Nancy, what is the clinical relevance of no performing anti-sperm antibodies?
So for if you are doing ICSI, I don't think there is any clinical relevance to it, because most of the centers nowadays I know are doing ICSI for all. But definitely, if you're doing IVF, you have to consider these factors. This is what I think.
What do you think the gold standard techniques, double DNA density gradient centrifugation and all, won't it help us eliminating those sperms and those molecules, those
Yes,
antigens?
definitely. Washing, sperm washing is also included in it. That if you want to do IUI for such kind of patient, you can use sperm washing with dilution effect to it. How do we do dilution effect? We take a few ml of culture media to the container, and then we send patient to collect the sample, and then we do double density gradient, and then after that we prepare the sample. And I think that will also eliminate or wash anti-sperm antibody from the semen, from the sperm.
Yeah, not only for IUI, even for IVF.
Yes, yes, sir. Yes, sir, definitely.
Well, thank you, thank you. Dr. Akash, I'll be sharing the screen to introduce the next speakers.
Thank you, Dr. Sanket. We had some wonderful talks about the sperm function tests. We have seen the vitality test over there, the ROS, the DNA fragmentation index, and the sperm antibodies. So moving further, the next talk would be based on the sperm aneuploidy test. And to take us further is Ms. Lalita Upadhyay, who has done a master's in biotechnology. She is a certified clinical embryologist from Pratiksha Infertility, Guwahati, India. She has 7 years of experience, and she is practicing at Pratiksha Hospital right now. She has got certified in advanced skill set embryology training from IIRFT, Delhi, India. So over to you, Ms. Lalita.
Thank you so much, sir, for the kind introduction.
You can share your screen.
So can you hear me?
Yeah, we can hear you. You can share your screen, please.
Sure. So is it open?
Yeah, you can put it in the slideshow.
Sure. So now I'm going to start my topic is on sperm antibody assessment.
Sorry to interrupt. Lalita, please go to slideshow.
Now is it showing?
No.
It's visible, but it's not full screen.
Yeah, make it full screen. Come to slideshow. You can see the corner button here. No, down.
At the bottom.
At the bottom, next to that 66%, there is one icon, right? Just click on that.
Bottom right.
At your right hand bottom. Yeah, next to that. No. Next to the 80%. Left of the 80%.
To the left
yeah.
of 66%.
Yeah.
To the left
Click
of
on that.
66%. To the left of it.
Left of it, yeah. Yeah, click
Go
here.
to view. Just ऊपर में देखना view लिखा है डॉक्टर ललिता. ऊपर just go on the top. There's something that is view after review. Go higher. View.
ऊपर.
और ऊपर जाओ. View लिखा है view. Just go above it.
आपका एरो जहाँ पे है ना उससे थोड़ा ऊपर जाइए.
Arrow से जाओ ऊपर. या स्लाइड शो मोड पे ले आओ. जस्ट उसके नेक्स्ट देखो एक छोड़ के स्लाइड शो लिखा है. जस्ट क्लिक ऑन दैट.
It's okay, Lalita, you can go ahead.
Yeah.
So my topic is sperm aneuploidy assessment, and the objective of the lecture, the basic of about sperm, what does sperm looks like, history of aneuploidy testing in sperm, and what is sperm aneuploidy, sperm aneuploidy test, is it technically feasible, is in implement aneuploidy assay in a clinical setting. Is sperm aneuploidy clinically relevant? What is the incidence of elevated aneuploidy? What causes sperm aneuploidy? Can therapy lower the sperm aneuploidy rate? And last is, what are responsible guidance for testing? So this is the basic about sperm. As we know that in the simplest of term, sperm is a male sex cell or gametes. Sperm cells are haploid, which means they carry half the amount of chromosome their typical somatic non-sex cell do. A reference from biologically classed chromosome are made up of DNA, which carries genetic information. When a sperm cell combined with the egg, which is also haploid, they join chromosome, making a diploid organism with 46 chromosome. This process is known as fertilization. This is why you inherit some trait from your father and some from your mother. Next is, what does sperm look like? Sperm cells are approximately 0.05 milliliter long. They are not visible to the naked eyes, but using a microscope, we can see the structure of sperm. They have a few parts. First, the head, second, midpiece, and tail. Head, which contain the nucleus where genetic material is carried, and midpiece and tail, which people it for swing and help beat the egg. Structure of a healthy sperm: tail, head, midpiece. What is the sperm aneuploidy? Sperm chromosomal abnormalities include structure and numerical abnormalities often observed with sperm aneuploidy. Sperm aneuploidy result from mitotic non-junction during oogenesis and sperm endogenesis, leading to abnormal gametes with deimosis and nidosome. Next is history of aneuploidy testing is sperm karyotype of. This is the first history karyotype of human sperm from decondition and metaphase arrest in hemostro ova. In 1978, interphase FISH in decoded sperm nuclei. Sperm aneuploidy test. Sperm aneuploidy test is a test to study the genetic factors of male infertility. It also for the involution of the presence of an abnormal number of chromosome, aneuploidy and diploidy in the sperm. This test evaluate the percentage of spermatozoa with chromosomal abnormality in a sperm sample. This is the process set methodology, which is the some steps are included. Sperm sample first you can take, then wash with culture media. Next step is Connolly solution. Next is sperm separate and declaration is sperm head, and hybridization, delegation, and analysis. Men with abnormal SET. Reproductive implantation failures more than 2%, IVF failure, recurrent miscarriage more than 2%, male factors, and miss causes previous of chemotherapy and radiotherapy. Is sperm aneuploidy relevant? 15% of recognized pregnancy result in SAB, 50% of these are chromosomal abnormalities, according to the study of 1984. Aneuploidies in gamete are predominantly from areas of meiosis during oogenesis. Fertilization does not select for aneuploidy sperms. There is some papers, Mercury, double metrizine make for 2 robotic translocation. Second is chromosome painting of chest, chromosome 8, 16, 17 by all sperm and blastomeres. Aneuploidy rates equal in sperm and first cleavage blastomere, no intrachromosome effect noted. Is sperm aneuploidy relevant? 15% of recognized pregnancies rate of FCN, 50% of these chromosome abnormal. Aneuploidy in gametes are predominant from errors of meiosis during oogenesis. Clinical relevance of aneuploidy. Sperm aneuploidy does not influence fertilization capacity, according to the paper of 1999. Correlation between sperm and PGS aneuploidy rate. The prevalence of sperm aneuploidy in higher in case of repeat IVF failure with male factor infertility. Last one is screening for the prevalence of a known translation and or associated aneuploidy in the male genome PIXI to ICSI in 2005 and 2008. Encoded evidence of anomalies. This is elevated chromosome 15 annulated transmitted to fetus carinin. For second one is 4 consecutive triosmic presence with elevated frequency of associated sperm. Last one is the other repeats 21 and 18 and X. Sperm aneuploidy and the resulting embryos studies. 32 couples enrolled in this study. Man may be age of 32. Tested chromosome 13, 16, 18, 21, 22, X and Y. T-test, sperm aneuploidy less than 20%, embryos on 35, blastomeres 628, chromosome abnormality 5%. In second, 2 to 2% 4.4%. With 25 embryos, blastomeres 380, and chromosomal percent of abnormality 63.5, and more than 4.4% embryos on 23, blastomeres 354, percentage of chromosome abnormality 73.5. Significant correlated between increased sperm aneuploidy and chromosome abnormalities in blastomeres. Next is chromosomal abnormalities reported in IVF embryos are correlated to sperm parameters. There is some parameters according to the Markey 2009. And gonosomal aneuploidy in 1,549 embryos study in May 2009. Chromosomal aneuploidy 1,054 embryos. They associate between sperm that indicates and the chromosomal status of pre-implantation embryos were assessed in 230 couples with a female partner younger than 36 years undergoing. and other is 295 cycles of pre-implantation diagnosis for aneuploidy, and 105 cycles had normozoospermia sample, and 135 cycle is presented with oligoactinoteratozoospermia, while the remaining cycle had the spermatozoa from the seminal tramp due to overstrict 29 cycles or non-overstrict azoospermia. One blastomere was biopsy from day 3 embryo and analyzed for chromosome XY 13, 15, 16, 17, 18, 21, and 22. Non-obstetric and angiospermia patient 69% what they find, and chromosomal abnormalities embryos was significantly lower in normozoospermia patient 55%, and NOA is 69 and with 134 cycle presented with oligoactinoteratozoospermia, and they find 60% of aneuploidy. From the recycle of all blastomere in 493 non-transferred embryo, 95% NOA embryos were coated mosaic. In conclusion, a severe male infertility condition could contribute to the generation of chromosomal abnormalities in the result of embryos. This may occur especially in NOA patient in which the high indices of chromosomal abnormality is mainly due to meiosis and genomal aneuploidy. Recurrent miscarriages in according to the Career 2003. Previous studies have indicated that sperm quality may be related to unexplained recurrent pregnancy loss. This study evaluated the degree of sperm DNA fragmentation using the Tune assay on sperm from 24 copies with unexplained recurrent pregnancy loss, and compared to sperm from 2 control groups, donor of known fertility and unscreened men from the general population. The percentage of sperm staining positive for DNA fragmentation was increased in the RPL group compared to the donor or general population control group. In the RPL group, no correlation was observed between some quality parameters and the Tune data. These data indicate that some RPL patients have a significant increase of sperm DNA fragmentation, which may be positive of pregnancy loss in some patients. How common event sperm aneuploidy was the clinical relevance. There is some points. Number 1, what is normal? Second, few systematic analysis. Third is standardization. Fourth one is import QC. Reported in incidence of aneuploidy. There is some studies where they include the reported instigation of aneuploidy, and mean aneuploidy rated 5 chromosome on 13, 18, 21 XY chromosome, A fertile normozoospermia. The percentage rate is 1.2, and B general population 1.8%, general infertile 2.7, teratozoospermia 3.6, poor embryogenesis 4.3, and recurrent miscarriages 5.1, several ultrastructure defects 14.8%. Incidence of relevant aneuploidy percent of sample, and total aneuploidy for 5 problems. So according to this, this is 5 chromosome 13, 18, 21 XY. The fertilization normozoospermia 0%, general population is 0.4%, general infertile 3.7, teratozoospermia is 3.9, poor embryos is 5.8%, recurrent miscarriages is 5.7%, severe ultrasound defects is 58%. Intra-individual variation. This is 2 studies. First one is TAMS and all 2009. Sporadic variation in all 10 subject over time, and second is single sample variation over trial. Second is studies rules, Rubies et al. 2005. These included intra-chromosomal difference, and second generally consist at 2 years, some variability at 5 years. Segregation analysis for translocation patient. So there is some papers, and according to 2008 paper Washdova, different segregation pattern and unbalanced sperm rate even with similar translocation. And in 2007, Chen et al. Different rate with similar reversion translocations. Third is 2009 Perry et al. Different segregation pattern for similar sex outcome translocation affecting chance of IVF success. And Yakut at 2006 sperm FISH unbalanced rate related to blastomeres. According to the 2008 Villan et al. Suggest high unbalanced rates, 60% is still conducive to IVF pregnancy, note high miscarriage rate. Breakpoint variability related to the variability in screen segregation pattern and subsequent unbalanced rate. What cause aneuploidy? Error in chromosome segregation lead to aneuploidy, a state where the number of chromosome in a cell or organism deviate from multiple of the haploid genome. Aneuploidy arising through chromosome segregation during meiosis is a major cause of infertility and inherited birth defects. So, progressive meiosis during spermatogenesis. So the process is the primary spermatocytes divide meiosis 1 into 2 secondary spermatocytes. Each secondary spermatocyte divide into 2 equal haploid spermatide by meiosis. Second, the spermatide are transformed into spermatozoa by the process of spermatogenesis. Chromosomal non-junction may result in aneuploidy. So here 3 form of non-junction. First is failure of a pair of homologous chromosome to separate in meiosis 1. Second one is failure of sister chromatid to separate during meiosis. Second failure, last one is failure of sister chromatid to separate during mitosis. So non-junction result daughter cell with abnormal chromosome aneuploidy. What cause non-junction? Non-junction occur when chromosome fails segregation during meiosis. When this happens, gametes with an abnormal of chromosome are processed. Recommendation mechanism. Biopsy sample number of crossover MLH40, and second is positioning in subtelomeric region. Third one is fidelity of synaptomal complex. The frequency crossover and MLH40, this is the two things, normal spermatogenesis and NOA in fertile men. So basically normal spermatogenesis man 49.8 plus 48 recommendation site range, and mean with NOA infertile 40.4 plus 6.1. Meiosis error are associated with male infertility, the formation of synaptonemal complex without a crossover event in increase in infertile men. So this is control observation azoospermia and non-obstetric azoospermia. Crosses over crosses over in subtumeric regions. So crosses over in the first division of meiosis. Prophase 1 crosses over occur between prophase 1 and metaphase 1, and it is a process where two homologous non-sister chromatid pair up with each other and exchange different segments of genetic material to form the two recombination chromosome sister chromatids. And subtelomeres are considered to be the most distal further from the centromere region of unique DNA on a chromosomes, and they are usually dynamic and variable meiosis of multi-chromosomal blocks of a sequence. This is meiotic errors are associated with male infertility. There are comparison P arm and Q arm, mean distance from centromere, the approximate crossover site on a P arm patient mean distance, and opposite side mean distance from the centromere, the proximal crosses over site.
Sorry for the interruption. Lalita, can you please rush up because we are already running out of time.
Okay, no issue, no problem. This one again.
If you can come to the conclusion.
Yeah, conclude actually. I think it is better to conclude. Your last slide is there. What is the conclusion? Yes, you can speak on that. There is no issue there. Yeah, you can speak on that. This slide is okay. You can speak on that.
Okay, so this is the final slide, conclusion and clinical recommendation. New advanced facilities of sperm aneuploidy testing analysis in a clinical setting. However, more data are needed in establishing different range, and further studies are needed on sperm separation techniques to select aneuploidy sperm. Further data are needed on specific sperm aneuploidy rate and embryo aneuploidy to assess relative risk. Recommendation is essential for normal segregation quantity and quality, and clinical screening may be useful in certain pathogens, which included. Thank you.
Thank you, Mr. Alta, for the talk. We are running short of time, so I'll just we will not be able to take the questions. To sum it up, we can say that the sperm aneuploidy test is one of the sperm function tests. You might ask, how is it a function test? Because the main function of the sperm is to deliver the chromosomes, the genetic material. So if there's anything going wrong over there, so that we need to look at it. The ratio of what constitutes an abnormal sperm aneuploidy test, they are quite variable and does not translate so very well into your clinical outputs. Main reason being, I think, is we are using a fish where it is where you have to kill the sperm and you can't use it on a live specimen again for your ICSI. First thing. And second thing, you're testing mainly for only 5 chromosomes. The sperm is made of 23 chromosomes. So obviously we are going to lag quite behind in terms of our sensitivity and specificity of this. Further updates are definitely required in this area and might open up new potentials for this. Thank you, Dr. Lalta. And now moving on to our next.
Actually, Akash, here I want to say something that Lalita, it is very wonderful talk. Actually, we decided to make it short because we want to express the want to show the practical things over here. But really, you made it very good presentation. It is good, but it is detailed about everything, each and everything you included in this slide. It is really wonderful, excellent talk. But we were thinking about the practical knowledge of that thing. So because of lack of time, we are sorry for that we interrupted you in between. Thank you.
No
Thank you very
problem,
much.
sir. Thank you so much, sir. Thank you.
Moving on to the next topic. Now we had dealt about something which is not seen sperm aneuploidy to something which can be seen, which is the Hyaluronic binding assay. So taking us through this talk would be Mr. Vakil Jagvir Singh. If you can have the slide of his CV, please. Mr. Vakil has done his master's from Punjabi University and proceeded further to do his PhD in clinical embryology with the institute which was associated with IIT Madras. He is presently practicing as an embryologist at Cooper Surgical. He has conducted multiple workshops which have dealt with the sperm morphology, sperm DNA fragmentation, advanced sperm selection, and vitrification. He is a member of ISAR IFS and Emerging Antimicrobial Resistance Society of India and conducted various training programs. So over to you, Mr. Vakil.
Thank you, Dr. Kash. So I'll share my screen. So just let me know when it's visible.
It's visible.
Yeah, perfect. So good evening, everyone. So we're going to discuss about Hyaluronic binding assay. So before going to the practical part, so we have a video demonstration which will play in the end of the slide. So just to give a brief, because we are short of time. So we all know the origin of hyaluronic acid, that we know that hyaluronic acid is a component which binds the cumulus cells together, and it is also an essential part of cumulus suspension. And we all know that cumulus suspension indicates that the oocyte is mature, and hyaluronic acid, which binds the cumulus cells, it also plays an important role in sperm selection, which we know that sperms also have the receptors for hyaluronic acid, and those receptors help the sperm to guide the sperm toward the hyaluronic acid, which is bound to the cumulus cells on the oocyte. And the studies have proven that these receptors are known as receptors for HA-mediated motility, and that also when the sperm goes inside the fallopian tube and it senses the hyaluronic acid on the oocyte, it goes toward that. It also indicates, I'll not go into much into this, we all know the receptors are there, and the sperm cell also have hyaluronidase activity, which we all know. The main thing is that the sperms which that bind to hyaluronic acid tend to show that these are normal mature sperms. So it is kind of a natural sperm selection by the oocyte itself, that the sperms which have the hyaluronic acid receptors, they will bind to the hyaluronic acid which is there in the oocyte cumulus, and then the sperm will raise the hyaluronidase, which will help to take that cumulus and goes inside near to the zona. So sperm that bind to hyaluronic acid have shown normal maturation, low DNA fragmentation index, and low frequency of chromosomal aneuploidies. And on the other hand, the sperm which does not bind to HA shows immaturity because they lack HSAP2 genes, and they retain cytoplasm in histones and have to show low DNA integrity. So there are some studies done on the just to see how the sperms which are HA bound, which are active toward binding the hyaluronic acid on the oocyte, and the sperm which are not active toward binding the hyaluronic acid on the oocyte, how they behave differently with different stains. If you can see, the HA bound sperms has shown better DNA integrity, better retention, does not contain any cytoplasmic retention, and does not persist the histones. So what is HBA? So HBA is basically a small glass slide coated with hyaluronic acid. So it is basically physiologically mimicking the thing which is happening in nature. So the basic biological process is the sperm goes toward the oocyte which has the hyaluronic acid, it binds to the oocyte. So to do the same thing, hyaluronic binding assay is basically a diagnostic application of TICSI, which we have already discussed in the first talk by Dr. Amita, that TICSI is a sperm selection technique in which we use hyaluronic acid to select a mature sperm. So hyaluronic binding assay is a diagnostic approach of that process. So we have a glass slide on which hyaluronic acid is coated, and it comes with a cover slip which has a grid like our Meckel's chamber. So we put a drop of raw semen sample on that slide, we put the cover slip on that and leave it for a few minutes. The sperm which are having the receptors, which are mature sperm, which are having the HA receptors, they bind to the glass slide, and then we count the number of sperms, bound sperms against the unbound sperm. So in the 10 grades, like we do in the case of Meckel's. The sperm with receptors with hyaluronic acid shown to have high DNA integrity with the studies which I'm going to show you further. And we do a scoring. So we count the number of sperms. So the studies has shown the patients with male patients with low binding score, less than 65% of sperm does not bind, it does not bind to the HA quoted on the slide, needs a sperm selection method for ICSI. In those cases, we can use PICSI, we can use MAX, we can use microfluidics or any sperm selection method at advanced level than the normal method. So if we see in a simple language, it is kind of the same thing what we are doing for DNA fragmentation testing, that we are assessing a sperm's DNA integrity with either SCD or with either tunnel with or SCSA. It is kind of that, but it is a quick method than all those tests. So it's a simple slide-based method ready to use. Just put a drop of semen, put a cover slip, wait for a few minutes, and then count the number of sperms and see how many sperms are bound. If the number of sperm bound less than 65%, you can opt that patient, counsel that patient for advanced sperm selection. Or if the binding score is more than 65%, you can proceed with the normal ICSI, that the chances of picking up a normal sperm will be less. So what are indications for hyaluronic binding assay? So unlike PICSI, same like PICSI, we cannot use PICSI in all the patients. We cannot use MAX in all the patients. We have some indications. Similarly, like that, we have indications for hyaluronic binding assay also. So patient with repeated failure, patient with male infertility issues, we already know, patient with recurrent biochemical clinical pregnancy losses, or in ICSI patients where you are planning to select a sperm selection tool. So if you're like, we are an all ICSI center, you want to opt for a test, you can go for DNA fragmentation testing, you can go for HB assay, you can go for tunnel assay as per the availability, as per the costing, or whatever is feasible to the center. And then we can decide in this patient, when I'm doing all ICSI, should I need a sperm selection method, or I can proceed with the routine sperm selection after DGC or swim up. So it is kind of making the natural thing. So we are using a natural ingredient which already exists on the site. So we are just mimicking the sperm physiology which happens in nature. So it is kind of a safe in that point of view. This test is just a diagnostic. You cannot use the sperm which are used on this slide. For that, you can go for PICSI or the other things which are available in the market. So HBS score correlates with fertilization, pregnancy, and cleavage rates. So there was a study done by Fan et al. In 2013 in which they divided patient into 4 groups: HBS score less than 15, 15 to 35, 35 to 60, and more than 60%. And they found out that where the HBS score was higher, then the more number of sperms were attached to the hyaluronic acid, which was quoted on the slide. The fertilization rates were better, the cleavage rates are better, and the pregnancy rates are better, but not, I will not comment on the pregnancy rate part, but the fertilization and cleavage rates are clearly better. Because going till pregnancy, there are so many other factors are also involved, but still their study shows like this. Then there was a more study conducted in 2005 by Jacob et al. That HA bound sperm showed lesser chromosomal repetitions. So low chances of chromosomal aneuploidies in HA bound sperm there was studied. Because it is kind of a natural thing existed, it's a kind of a natural sperm selection existed in nature. So they just try to mimic that, and they found out that these things work because HA bound sperms are known to have mature with respect to the DNA, with respect to morphology and the physiological function. So I'll straight away move to the video presentation that how we do hyaluronic binding assay testing. This is just a slide which is coated with hyaluronic acid, same like the PICSI dish, which has 3 dots of hyaluronic acid. Here in HB assay, we have a whole surface. The red area under this is coated with the hyaluronic acid. And the cover slip which comes with the grid looks like this, the same like our macular chamber. If you can see the sperms which are bound and the tail is wiggling, if you have used PICSI, you know this phenomenon, you have seen already. But if you haven't used PICSI, so the sperms which are mature, they will bind to HBA, hyaluronic acid, and the tail keeps wiggling. Yes. These are the sperms which are mature with respect to the hyaluronic acid receptors. This red circle, under the red circle is the hyaluronic acid coating, and under the blue circle of the cover slip is the So you just have to take, you can either use raw semen sample or you can use prepared semen sample as well. Just put a drop of raw semen or prepared semen and 10 microliter drop on the glass slide. Put the cover slip on. and then wait, leave it at room temperature and wait for 10 to 15 minutes. So always make sure that when you're placing the cover slip, avoid the bubbles because bubbles can interrupt the binding. So just keep it at room temperature for 10 minutes, and after 10 minutes, just assess it under the microscope at 20x. So you can just simply see the sperm which are motile, which are moving from one place to another place. These are the immature sperm, and the mature sperm which are bound. They just keep wiggling that in at one place, bound to that sperm. You just have to count the number of bound sperm and number of unbound sperm. You can count total 200 sperm, like the same you do in some method counting. 200 to 400 sperms. You can increase the efficacy by 2 people making the test, and after that you can just simply take the percentage of bound versus unbound sperm. So more than 65%, if the HbA score is more than 65%, then you can simply proceed with the routine experiment without any indicator for the PIXI or MAX or something. If the result is lower than 65%, then you should use a method as recommended by the literature that you can go with a sperm selection method so that we have a method with respect to the DNA integrity. So to summarize this, so hyaluronic acid selection basically reflects that oocyte is also mature and the sperm is also mature, and the mature sperm will also always go towards the hyaluronic acid bound, which is there on the mature oocyte. So HA bound receptors on the sperm and HA binding sperm flex sperm maturity, reduce apoptosis and fragmentation. Sperm binding indicates sperm functionality, and higher binding associated with higher embryo cleavage, pregnancy rates, and lower chromosome replication. And still the studies are going on. This is basically, if I have to summarize in one line, so this is a diagnostic PIXI, which we're doing just to assess that the patient needs an advanced sperm selection method or not. It is quick, easy, reliable, and cost effective, and also time effective. So thank you, Dr. Akash, over to you. I hope I finished in time.
Thank you, Mr. Vakil. Yeah, thanks so much for going it through quickly. So hyaluron binding assay, like we have said, it is a short PIXI in short, which is at the diagnostic stage, and we'll not go into the details of pregnancies and dose.
Yeah,
I think that would be a topic for all together. Yeah, Dr. Sanket.
Yes, thank you.
Yeah, I have one question for Vakil.
Please.
It was very fun, neat presentation, Vakil. So I have one basic question for you. Yes, see, firstly, I'm pro HbA, but I want to know from you. See, if you look at it, it's a one binding technique, right? So if you consider the entire spermatozoa, this is just one aspect of looking at it, the quality of the spermatozoa. So how do you defend this?
So even when we're doing PIXI also, so it is always recommended that binding is one thing. Binding is one thing, but you have to keep the other factors also an aspect. I'm also a very pro morphology guy because I've worked with Daniel for a long time. I'm very pro morphology guy. So I would my selection criteria for a sperm will be binding, morphology, and motility. So at least 3 parts. That is my personal, because that is how I used to work. The 3 parts: binding, morphology, and motility is one thing. And the other thing, because motility tells me that it is progressive motile, it goes in a same direction, so there's chances that's normal. Morphologically normal, that means this functionally is better. But just to know exact that is functionally mature or not, then the binding. So I will go for 3 things: motility, morphology, and binding.
Do you mean to say that it is based on partially chemotaxis thing?
I would say yes, it is kind of a partial chemotaxis because there are 3 things. So I'm covering rheotaxis with my progressive motility.
Right.
Okay. Then I am covering kind of a thermotaxis with motility and some amount with morphology.
With the conditions where you're providing.
Yeah. And then I am covering chemotaxis with the HA receptors on the sperm head. Kind of a chemotaxis, yes.
Partially, yeah.
Partially, yes. Other factors are also there, but one of the factors of chemotaxis.
I have one question.
Forget about geotaxis and all. We'll concentrate only on these 3 as of now.
I think your geotaxis and
I have one question actually. May I, Akash?
Yeah, Paresh, please. Yeah, please, Paresh, go ahead.
So my question is, just think you have a two sperms which has a good morphology and motility, but not binding. And another sperm is lower motility and morphology is not good, but there is a binding. Which sperm will you select?
So morphology, which morphological abnormality is there? Then I'll go to that.
No, I will say excellent morphology with good motility sperms.
No, sir. In the normal morphology one, which
Normal
morphology?
normal morphology. Because actually, I am using sperm slow.
Yes, sir. Yes, sir.
Yeah. In sperm slow, I have found sometimes that sometimes there are good motile morphological sperms.
Yes,
they
sir.
are moving very fast, but they don't have a binding capacity. And at the same time, I'm finding the sperms which are the vacuoles and head abnormality, and they have a binding effect. I don't know which sperm we have to select.
Good question, I think.
Also, it is like, what is your order? What is your criteria out of the
What
things
is
you
your
have
order?
told?
Yeah, right.
That was my question also. Like, what
Yeah.
was the order? What
You
you
are-
go for? Whether it's motility
sir,
morphology versus your binding.
No, so that is why that is what I said. So one thing actually the tricky thing which I faced, that sometimes, as Dr. Paresh said, a morphologically normal looking sperm can have a bad DNA. When we do, even when we do SCD or tunnel also, when we see SCD especially, we see there is a good morphologically normal sperm, there is no halo. And then there is a normal morphology sperm, and sometimes a double tail sperm, there is a very good halo. But there are so many factors, so I would make a balance with which morphological abnormality is there on that particular sperm. First, that if it is a head morphological abnormality, then I would rather avoid it for the initial. But if it is binding, that somehow it also shows me that it is matured, it has the HA receptors. And if I go biologically, HA receptors are only present on the sperms which have genetic maturity, which have a maturity with respect to the PA20 genes with the hyaluronidase activity.
But to be frank, even a minute change in the temperature might
Yes,
shut down
yes,
receptors.
Yes, even when we do
as well.
If you do the HA bounding test at 37, you may found that there are less sperm binding to HBA, hyaluronic acid, because at a high motility. So that is why they always recommend to do it at room temperature, so that this sperm motility has to be in a normal phase.
So what about ICSI during PIXI? How do you select them? PIXI, we are not doing on that room temperature, no?
So
PIXI is advised at room temperature.
PIXI is advised at room temperature, sir. Yes.
But from selection, from that place to selection, then you have to change the dish. It means.
No, sir. No, you don't have to change the dish. You can keep the same PVP drop in the particular dish also. You can put a PVP drop in the particular
temperature, room temperature, maybe room temperature is 23. Then.
So room temperature has to be at 24 to 26 to 28.
So then you are using 24.
It's there in the SOP.
How it is recommended? That is my question actually.
You might shut down the receptors.
Yes.
Because the doors, instead of opening, they might close. That's what,
anyways, fine. It was wonderful.
Yeah, I think there are some practical difficulties associated with the PIXI. And the loading, what you have to do, if you remember the discussion we had, the amount which you need to get over, that's very, very important when you use sperm slow and PIXI. It's more related to the absolute number of sperms that you are going to load, because it can have a cascading effect on that as well. So I think we want to go more into this discussion, but we are falling short of time. It opens up
next time, Vakil, you have to prepare everything now.
Yes, sir, definitely,
yeah.
sir, definitely.
So I think thank you, Mr. Vakil, for that nice presentation, and we'll move on to our next topic, which is being done by Mr. Hardik Seth. So we have seen the unseen, what can be seen. So now he wants to eradicate the human error part of it, and where we come into the automated semen analysis, how automation works over here. If you can have a slide, please.
Okay, can you please share that slide?
Yeah, sure, sir.
So Mr. Hardik is working as an embryologist and scientific officer at IKDRC ITS. He has done his master's and in life sciences, and followed by master's in hospital administration as well. So over to you, Mr. Hardik. You can start your presentation.
Just a minute, sir. Am I audible, sir?
Yeah, you're audible.
Yeah, okay, thank you. Good evening, respected seniors, all the IHERA members. And thank you. I'm personally thanking the IHERA team for providing me this opportunity to present in front of you, rank and file people. Today, my topic of discussion is automation in semen analysis. What is automated semen analysis? The automated word itself meaning the use of the machine. So it is basically an automatic or the semi-automatic system which utilizes the modern computer and advanced image processing techniques for clinical analysis of sperm motility, morphology, vitality, and DNA fragmentation. Based on the WHO standard criteria of WHO 5th manual, the system analyzes the sperm characteristics and generates the accurate parameters of sperm. The whole procedure is very fast and provides the several and important information that are vital to the current scientific basis for the male reproductive ability. Now, after this, the first question rises in our mind is that how does the system works? What functions it includes, and what parameters it works on? So for this, the user firstly, after opening the software, the user firstly have to feed the patient information data, in which the user has to feed the patient information, patient name, ID, the volume of the sample diluent which the user has performed, and the facility name also is important, and the chamber in which the sample is loaded, the slide chamber in which the sample is loaded. Once the temperature reaches to the 37 degrees Celsius, here you can see the first image, where in the left the 20.8 degrees Celsius is written. That temperature has to be reached to 37 degrees Celsius. Then the preloaded slide should be loaded into the loader, and the analysis has to be started. So what the basically system contains is the microscope, which is attached with the high resolution camera, which takes the video of the semen sample, and the sperm's image are being grabbed. Now the count and motility assessment. In the count and motility assessment, here is the first image where you can see the ongoing analysis is in progress. And this is the animated image where you can see that the consecutively 30 images are being taken in less than 0.5 seconds, or say in less than 1 second. So you can imagine how fast the CASA system works and how accurately it works. Now this is the image where the assessment by the CASA system shows the different number of different sperms in different colors, by indicating the cyan or the green colors indicates the motility. The pink one shows the slower motile sperms, and the red one are the statics. Okay, the software then generates the. I'm extremely sorry for the technical inconvenience. The software then generates the count and motility as per the WHO guidelines in million per ml. So these are the images of the reports that are being generated after the analysis has been performed. So here you can see in the first image that the motility in million per ml, the million per ejaculate count is being given. There are other motility parameters are also there, which shows the progressive motility, total motility, and the non-motile sperms are also there. There is also graphical representation which is being given by the CASA, which shows the progressive, non-progressive, and immotile sperms in different colors, like green for the progressive, and yellow and red are for the non-progressive and the immotile respectively. Now the playback function. What is the playback function? The user can use this playback function, which is being provided by certain CASA systems. This playback function can be used by the user just to check whether what the CASA system has analyzed actually, and this can be taken after the analysis has been performed by the CASA system. Now these are the kinematics which is being included by the CASA system for the reporting. That is the straight line velocity, which is the distance between the first and last point on the sperm tract. Curvilinear velocity is also there. The velocity which is along the sperm tract is being measured by summing the distance between the sperm head position in each frame divided by the elapsed time. The average path velocity is also there. There is a sperm linearity is also there. Sperm straightness is also there. Now this is the video of a CASA system which uses the raw semen sample, and here you can see the report is being generated which shows the million per ml concentration. The morphology was also being shown by the CASA system. And here in the right corner, you can see those white circles over the sperm that are indicating the abnormal morphology of the spermatozoa. Now, this is the image of the CASA system generated after the analysis that indicates that this sperm is morphologically abnormal. So this type of sperm could be avoided while doing ICSI or any procedure. Now, this is the morphology report. The CASA system also generates the report for morphology, where it gives the result in person normal or abnormal. DNA fragmentation. CASA system also performs the DNA fragmentation. As the ROS level rises, the DNA fragmentation is the level of DNA fragmentation high. A high level of DNA fragmentation in sperm cells may represent a cause of male infertility that conventional examinations, sperm concentrations, motility analysis, morphology assessment cannot detect. Even at the higher magnification, like 1000x magnification, a normal looking sperm may have the DNA fragmentation which cannot be detected normally. Elevated levels of DNA fragmentation will significantly compromise the possibility of a successful pregnancy. Here you can see the report of the DNA fragmentation having the big halo, small halo. Big halo is indicating the normal fragmented, and the this is the fragmented one. There is also graphical representation of each and everything, so that can be used for the analysis purpose and research purpose also. So why to move to the CASA then? Despite the efforts of the WHO to standardize the manual semen analysis, the technique still lacks the reproducibility and accuracy. And this inaccuracy is associated with the counting, statistical errors, poor sample handling, lack of consistent adherence to the protocols, and workload stress to the technical personnel. This is further compounded by instrumental variations varying in protocols and nature of time-sensitive biological samples too. The accuracy and the precision can be achieved by eliminating human errors, adhering to an effective and standardized protocol, and assessing very large sample size. CASA, why we need to choose the CASA is because it provides the objectivity by which it reduces the interpersonal variations. Accuracy, by this it ensures the indifference in sperm concentration, motility, and morphology estimates. Sensitivity, that detects even the slightest movement in the sperm. Precision by standardizing the observables with the higher limit of acceptance and reliable reproducibility. And at the end, the records, the user have the option to have a huge data bank for the patient record, which can be used for research purposes and record purposes. And these are the support evidences which indicates that the validity and reliability of the semen analysis are now taken. So in the end, I'm concluding the presentation with the scientific evidences suggest that the CASA system is a valid alternative for the evaluation of semen parameters in clinical practice, especially for sperm concentration and motility. Artificial intelligence-based CASA devices promises to offer higher efficiency of the analysis and improve the reliability of the results. However, further technological improvements are required before these devices can one day completely replace the human operator. Thank you.
Thank you, Mr. Adik. It was a nice talk, crisply up to the point. So as Evan said, CASA holds a promising technology. One question which I would like to ask you is, what are the various systems which you have used and what is the compatibility? Like how do they closely
I beg your pardon, I wouldn't be able to hear you clearly. Will you please be louder?
Can you hear me now?
Yes, yes, sir, I can hear you.
Yeah. So what are the various CASA systems that they use? What are the major drawbacks of that in terms of which parameter you feel it has been most of drawback is there in terms of like count, motility or morphology? That would be the first part. And the
Okay.
second part would be, what are the different CASA systems which I've used personally, and what do you feel is the major drawback and advantage? One point each.
Pardon, sir, pardon?
One point each for the major advantage and disadvantage of CASA system.
The major advantage is that it minimizes the workload of the technical personnel and gives the accuracy, but it still lacks the standardization. Like whenever the user has to use the CASA system, it has to standardize the CASA system manually first. Say for instance, the dilution factor. The dilution factor is most important while counting or performing the analysis in the CASA.
Okay, and which parameter do you think correlates most and correlates the least? Count, motility, morphology compared to manual.
Sorry, sir?
Like you have three parameters majorly when we do semen analysis, right? Your count, motility, and morphology, right?
Yes, sir.
So when you compare manual semen analysis versus
Yes,
the
sir.
automated CASA systems, which is the parameter which correlates the best between the two, and which is the one which correlates the least?
The variability in the low concentration and high concentration, sir?
Yeah, in terms of concentration, it's
Yes.
much more accurate, but at the extremes of it, it doesn't perform well when it comes to very low concentration or very high concentration. Yes. And what about motility then?
So with motility, I have no idea with the motility, sir.
It should be the same.
It's the same. It's same at the extremes. You know, after all, these are machines, right? So
Yes,
they don't
yes, sir.
correlate so well when you go to the extremes of it. And like you have very rightly said, they still require quite a few modification, tweaks to it. It's all not up to the mark, because whatever I have seen, it is much more variable in terms of motility when you compare to the human factor. It might be that the human eyes are trained differently. It's not that the system is wrong. It might be
Yes,
just
sir.
the human eyes are trained differently to pick it up. So that's why we are getting more variability in terms of motility. And the method by which these systems pick up the count and motility, they are different. Like they use the optical effect. Sometimes they'll be using the electrical resistance. So different CASA systems use different kind of technologies, how they calculate this count and motility. So that's why probably these CASA systems are variable, and there is variability when you especially compare with the manual systems, like your manual semen analysis.
Yes, sir.
Any other questions, Ved sir?
Sir, you are on mute. Yes, sir, you are on mute.
Yeah, so I think we have to wind up because already 8:30 is gone, and
Yes.
all the delegates are going out. So I think it is wonderful. Actually, I like today. I like one thing that all the presentation was very up to the mark and excellent, and we discussed lot of things, lot of practical things. One thing we have to do in our next upcoming webinar, we have to keep the practical points in it. Main focus should be on practical points, not the theoretical points, because this is doing this webinar was the practical discuss the practical points. So, but overall, it was very good, and we discussed a lot of practical things amongst us. And thanks, Sanket and Akash, and of course, Praise and Vakil. We discussed so many things in between, and I hope people also like these things. And after all, everyone want to know the tips and tricks. What is the basic things to upgrade their skills? With that, I thanks all of you for joining us. Thanks to Cooper Surgical for supporting this webinar and all my teams. Thank you.
I think we must do a part of this, which is mostly of the discussions.
Yeah.
Because a lot of people are waiting, and even the speakers are waiting for the discussion.
Yeah.
Really wonderful.
It's the discussions which we'll remember more at the end of the day, and these points which we take home. And it was really nice interacting with all of you and seeing the wonderful presentations. Definitely, we can pick up from each other's mutual experiences. Definitely points which we can pick up. So thank you everyone for that.
Thank you.
Thank you, Dr. Ved. Thank you, Team Ahira. Thank you, Dr. Sanjay. Thank you, Dr. Agarwal. Thank you, Dr. Paresh, for moderating the session. And
thank you so much.
Thank you again from side of Cooper Surgical. Yeah, looking forward to see you again. Yeah.
Thank you.
Bye, bye, bye.
Bye, everyone.
Are we offline?